Hello, good morning, everyone. I'm Soumya, a postdoc in Anna Wang's lab, and it's my honor to chairing the session today. So welcome to the third day of the Australian Astrobiology meeting 2023, and sadly it is the last day. So we have 4 talks in the morning session, and with the whole day is like pretty much same as like first day session. We have 4 speakers, and I just want to remind our speaker that our invited speaker have 30 minutes in total time for talk, 25 minutes for presentations, and 5 minutes for Q&A, and oral speaker have 20 minutes total. So without wasting time, let's introduce our first speaker today. It's Dr. Nick Green from University of Otago, Department of Chemistry. Dr. Nick Green is interested in chemistry of molecules of life, including nucleotides, peptides, and protein molecules, and their potential synthesis of the molecules in the primordial earth. So let's welcome Dr. Nick Green.
Very much. Good morning, everyone. Oops. Congratulations on making it to the final morning of the conference. And yeah, thanks for having me along. It's been a really fun few days. Good to be back in the conference swing of things. So yeah, going to show you some chemistry today. I apologize in advance for the chemical structures for those of you that aren't chemists. But of course, it's nice to start a talk with this philosophical question. And of course, you know, in the next 25 minutes and of course, in the past 3 days, we haven't been able to quite answer this question, but hopefully what I'll show you today will help convince you that, you know, there's different ways of approaching the problem. We can look sort of in a backward sense and try to trace what we think must have happened by working backwards, but we can also just explore chemistry and see which chemistry is fruitful and therefore perhaps make inferences about what chemistries may have had to occur on the primordial Earth. So just with respect to that process, of course, we can think backwards and sort of define the geological conditions at the beginning of the Earth, but we can also think in a chemical sense about what the chemical processes and what the constituents of these mixtures need to be to get to something that we can recognize as biological eventually. And so we've discussed what we would recognize as a biological system a little bit already, the sort of chemoton, the basic unit of biology. So if we look more specifically at that biological system, we know that it's composed of various classes of biomolecules that we can sort of, from a simplicity perspective, just roughly divide. So we have a simple metabolic cycle that harvests chemical energy. Of course, we need lipids to form a membrane. We've had some interesting discussion about them already as well. Amino acids, in modern biology at least, provide the catalytic machinery of a cell. And of course, at the center of all of these systems and controlling these systems is the genome, which stores information and can also supplement the catalytic systems in that cell. So my background for the last few years has been mostly working on the genome side of thing in the RNA, DNA type world. And so you can see there's been a bit of attention in there. But today I'm going to talk to you a little bit about this amino acid side of things. Just some recent work that we published on amino acid synthesis in Formamide as a prebiotic solvent. And just to talk about the sort of overall philosophy, coming back to what I hinted at the beginning of the talk, is that we're really exploring, you know, we're not limiting ourselves exactly to the molecules that we think are necessary, but once we have a few of those and we sketch out what we think are the basics for being necessary, then it becomes about sort of expanding what we can think of as that primordial soup. And then if we realize that certain molecules might be connected to each other constitutionally or via their synthesis, their abiotic synthesis, then perhaps investigating the relationships between those molecules and how they interact, that might lead to the increase in organization that we need to see to go from a primordial soup, which is just a sort of disparate mixture of molecules, into something as organized as a biological system. So amino acid synthesis, one of the cornerstones of prebiotic synthesis is, of course, the Strecker amino acid synthesis reaction. So we know in the Miller-Urey spark experiments that this is the mechanism that's responsible for generating amino acids. And there are other variations about thinking of using this chemistry to generate amino acids in aqueous solvent. And how this reaction works is you take a simple aldehyde. So Steve showed us a lot of simple aldehydes yesterday. You take ammonia and cyanide, you mix them together, and the first intermediate that you're interested in really is the amino nitrile, which is this compound here. And that, in the presence of water, can undergo hydration and then hydrolysis of this nitrile group to give us amino acids. So it seems pretty simple. And indeed, amino acids are relatively simple compared to nucleic acids, for example. But even though this is a widely accepted and a really useful way of making amino acids that seems prebiotically plausible in many different contexts, it's not without problems. So it requires a high concentration of ammonia. There's been some really nice work in the literature quantifying those rates, and it basically comes down to this pH-independent relationship where, of course, the concentration of ammonia is still related to the pH of the solution. But if we take an example and we just use neutral pH, then to push this reaction over towards the right, you need really high concentrations of ammonium ion in your bodies of water. So perhaps that's problematic. It's a pH-sensitive reaction in general as well. It works better at higher pH where you have higher concentrations of ammonia. And also, it's sensitive to carbon dioxide. We know that carbon dioxide, well, we don't know, but we suspect that carbon dioxide was a major component of the primordial atmosphere. And so what happens is it intercepts these intermediates here and generates these high Dantoin compounds via a type of chemistry called Bucherer-Berg synthesis. And these don't generate amino acids. They generate these relatively difficult to sort of convert to amino acids, high Dantoins here, and carbamoyl amino acids. So even though the Strecker reaction is great, it does have these limitations. So we started looking at amino acid derivative synthesis in the context of a different solvent. So that chemistry that I just presented, unsurprisingly, we've focused on synthesis in water because we know water is a necessary ingredient for life. I think Martin was saying yesterday, one of the key sort of ideas in Origin of Life, research towards the origin of life is that the conditions before life that generate the conditions necessary for life might be completely different. And so one of those differences could be the prevalence of this solvent, formamide. So it's an organic molecule. And the reason it's interesting is if we consider the source of nitrogen in biomolecules, nitrogen always has this oxidation level that corresponds to these molecules down here. So cyanide and ammonia. And that's also the same as formamide. And so cyanide we can think of as a really key building block that was delivered to Earth via different methods, exogenously and also via production of high impact, high energy impactors and so forth, processing nitrogen that was already on the Earth and converting it to cyanide. And cyanide is really useful, and I'll show you that in just a moment. But it's this relationship. If you accept that cyanide is useful, then obviously it would be nice to investigate the properties of formamide as well. We're certainly not the first people to do this. Jim's been involved in this. Steve's been involved in this as well. And so, yeah, we thought, given these properties, perhaps having accumulated formamide and considering the sort of chemical and physical robustness of this compared to its relatives, cyanide and ammonia, perhaps we could investigate amino acid synthesis chemistry that was not just looking at formamide, but looking at the co-occurrence of these three molecules that are related via this hydrolysis or this hydration and hydrolysis reaction. So basically the summary of that is to say that if we accept that cyanide is a useful and plausible molecule, then we may as well investigate the chemistry that would occur from mixtures of these compounds. So a little bit more detail on how that might work. So cyanide is really useful in generating the types of aldehydes that Steve was showing you yesterday via this homologation type reaction, where a photochemical homologation reaction that's been developed in John Sutherland's lab. And so cyanide is related to these aldehydes, which are really useful building blocks for building the nucleic acids, the sugar component of nucleic acids. But meanwhile, via this relationship that I just showed you, cyanide can also be processed to formamide. And these components, combined with the aldehyde, are also the components of amino acid synthesis. So rather than just isolating different components of this biological system that we're aiming for, we can start to imagine how the same chemistry, when it's nudged in slightly different directions, might give us all the necessary components rather than just one or another of a biological system. So we started with a simple question. We didn't start with the aldehydes. We wanted to get some sort of controlled background reactivity. So we started with aminonitriles, which are that first important intermediate in the strechoamino acid synthesis. And we just asked, how do they react in formamide? So for a synthetic chemist, this is a fairly easy reaction to predict. And it turns out that when you have unreactive side chains on your amino acid precursors, this reaction goes exactly the way you would predict it to, and you get a transamidation reaction, an acyl substitution at this carbonyl group. As Steve pointed out yesterday, carbonyls are reactive moieties. And basically that transfers this formyl group onto our aminonitrile. So the aminonitrile becomes an N-formyl aminonitrile. So it's not a particularly remarkable transformation, but we'll come back to why these products might be useful in a moment. But now I just want to take a look at this one example. So when you start with glycolaldehyde, so this is the C2 aldehyde that featured prominently in the Formose reaction yesterday. So that's where you've got a CH2OH side chain. This reaction doesn't go in a nice, easy way. You get this N-formyl aminonitrile to start with, but then the yield starts to decrease over time. And we figured out, we fished out the compounds from the reaction, and we figured out that alongside this formylation product, we're getting two other major products. And so one of them is this compound here, where we've basically installed 2 units of formamide onto our starting material, and it was a little bit interesting. But the one that really piqued our interest is this compound here. So this is N-formyl dihydroalanine nitrile. So just how we get those names. So this is an example of alanine. It's an amino acid. So it's got this acid functionality. The aminonitrile has the nitrile. And remember, these are related by just processing in water. And so when we talk, we have these FO, that means formyl. So these are the N-formyl nitrile derivatives, just to help you out with the terminology. And so in these reactions, we're getting up to around 20% of both of these compounds, which, that's a major sort of component of this mixture. And so we wanted to investigate how these were forming and whether they could be prebiotically useful in a prebiotic context. But of course, we were cheating. We were starting with a preformed starting material. So now we went right back to that idea that I talked about at the beginning. If you accept the relationship between aldehydes, cyanide, ammonia, these sorts of compounds, what happens when you just mix them together? And so we found that this really cleanly pushes this reaction over to the right to generate our N-formyl aminonitriles. Just like I showed you on the previous slide, we get pretty high yields of these where you have uninteresting side chains on the aminonitriles. But again, in the case of serine, low yields. And that's because of the same thing happening. And we're forming these 2 products in pretty reasonable yields, 18, 25% in our one pot or direct reaction that proceeds from glycolaldehyde, cyanide, and formamide. And I should note that this was the sort of final reaction we did. There was an intermediate reaction where we also had ammonia present in these reactions, but it's worth noting that we haven't got any free ammonia in these reactions. And so that is a potential real improvement on the limitations of the Strecker reaction, where you do need those high concentrations of ammonia. And of course, the nitrogen in this reaction is coming from formamide itself, which, as I mentioned, is maybe a more robust source of nitrogen than ammonia itself. So this is kind of a contrast to the things that Jim was showing us on the first day. So in this type of reactivity, we're really looking for selectivity, okay? Because, we had that discussion about how if you start to branch in too many directions, then it might be difficult for function to emerge from those particularly complex mixtures. We want to limit the complexity. And so this is an NMR spectrum of this reaction, just to demonstrate that, this is a clean and a real reaction. So these peaks in red are this interesting dihydroalanine nitrile derivative here, and the peaks in purple and blue are the other 2 compounds that I've mentioned here. And you can see they really are the major components that you get when you just mix together aldehyde, cyanide, and heat them up in formamide. So what's happening? Well, we start with glycolaldehyde, and it undergoes addition from formamide to form a type of intermediate here, an N-formyl hemiaminal, and that can undergo a reaction with cyanide that's very similar to the Strecker reaction, except instead of using ammonia, we're basically using formamide as our amine source. And so that gets us to our first intermediate. So we can really call this Strecker-like chemistry that's happening in formamide. There's nothing particularly crazy about that type of reaction. But the cool thing that happens when you've got this serine side chain is that when you're in Formamide, you can also formulate, again, just by heating, nothing fancy or crazy. You can formulate that side chain, which activates it towards elimination. So that side chain becomes eliminated and it ends up being an alkene to generate our dihydroalanine nitrile derivative here. And this is a cool reaction from a chemical sense. And it doesn't necessarily mean it's prebiotically relevant, but it's an interesting thing to know that this also maps the biosynthesis pretty well of dihydroalanine, which I should have said is an important biosynthetic intermediate in its own right, but also particularly in the biosynthesis of cysteine, which is an extremely important amino acid. Meanwhile, this dihydroalanine, this alkene, is activated towards addition at this terminal beta position here. And so it can undergo a second addition of a formamide molecule to that beta position to install this compound here. And again, this is a related, if you take the amino acid version of this is another metabolite that does feature in biochemistry. So there's some, tantalizing hints that maybe we're onto something that could be useful from a prebiotic perspective. But again, coming back to that philosophy, we're sort of fleshing out this prebiotic soup and trying to understand what type of chemistries are going on. And so we went back to our dihydroalanine nitrile derivative and just thought we'd test its reactivity in the presence of a range of primordially plausible molecules. So a simple reaction with cyanide, for example. And before I get into the details here, what we discovered was that there was a dichotomy of reactivity between this alanine nitrile derivative in water versus in formamide. And so that shows us that, we can access different types of chemistries by nudging that chemistry in a different direction once again. So perhaps that's a useful way of approaching the complexity that we need in a selective manner under certain microenvironments. So when we take cyanide and add it to this molecule here, what you would expect if you've done some chemistry is to get addition of cyanide at the beta position there. And indeed, that's what you get if you do this reaction in formamide. However, if you change the conditions slightly, then basically under more basic conditions where you haven't neutralized the sodium cyanide, then you start to get in situ hydration of the nitrile group to amides here. And basically that's just helping this reaction along to get to its amino acid derivative eventually. So that was kind of interesting, but what was remarkable in contrast to that was when we did this same reaction in water though as the solvent, we got something completely different. And it was this cascade reaction where instead of just a single molecule of cyanide adding to the alkene of dihydroalanine, we've got 3 molecules of cyanide adding to generate this interesting substituted imidazole derivative. Again, it's a pretty clean reaction, so perhaps it's telling us that this is a product of the inherent reactivity of these types of mixtures. And this is a new compound in a prebiotic context, so we're investigating potentially catalytic properties of a molecule like this, which having an imidazole functionality makes it a potentially really interesting compound, analogous to histidine, for example, which is an important amino acid in catalytically active sites of proteins. What other chemistry did we do? Well, we tried some other additions. If we take imidazole itself, which is just a model nucleophile, then it adds very cleanly to that beta position, unsurprising chemistry. And now if we get into some interesting chemistry, if you take a component of volcanic outgassing, hydrogen sulfide, dissolve it in water, sodium hydrosulfide, under various conditions, we got these different molecules here, which are all related to cysteine. And so the one that's most obviously related, this is just the formulated nitrile derivative of cysteine itself. And so really what we're showing here is that if you take this compound, which we think is a very reasonable molecule to expect, where you have co-occurrence of aldehydes, cyanide, and formamide, and you also have some hydrosulfide in that region as well, then you get to cysteine essentially, or the precursor of cysteine. And this was the first really, I think, plausible example where we're showing that cysteine could be a prebiotically available amino acid rather than the typically assumed late biosynthetic invention. Cool. So I guess the summary of what we're doing here is to just follow the chemistry. I guess that works if you're a chemist, maybe not so much if you're a geologist or a space scientist, but we've enjoyed following the chemistry and you discover interesting reactions that you don't necessarily expect. And so really we took what was already an important system of prebiotic chemistry based on cyanide, and showed that if you accept that these reactions are important, then perhaps you should accept that you're also going to have these molecules constituting parts of the primordial soup, and therefore perhaps playing a role in this important emergence of organization. So I've got time to just discuss one more sort of aspect of this chemistry that perhaps makes it useful, and that's in the context of biological or protobiological peptide synthesis. And so the way that biological peptide synthesis happens, of course, is mediated by the ribosome. And what the ribosome does is it brings together an activated amino acid. It brings it next to an already growing peptide. And it's because you've activated the end of that peptide that the amino acid is able to join. And so peptides grow in this direction in biology, from the N terminus to the C terminus. And the important thing is that you have this activation, this X group, and that allows this reaction to proceed in a thermodynamically favorable way. And essentially, if you want to simplify it, all the ribosome has to do is bring them together, and then the chemistry takes care of the rest of it. However, there's a problem with this type of synthesis, and it's the same problem if we try to replicate it in the lab. And that's if you activate the end of a peptide, when you get to this special stage in peptide growth where you have 2 amino acids. So you've started with 1, built 2 now. You want to go on to 3, 4, 5, up to 100 amino acids, like you would have in a typical protein. When you activate the C terminus of a dipeptide containing 2 amino acids, rather than undergoing addition from a separate amino acid, it has to compete with this intramolecular process to form this easy to form 6-membered ring, which is called the diketopiperazine. And this is a real problem. And the way this problem is solved in the chemical laboratory is by using N-protecting groups. And it might surprise you to find out that bacteria biosynthetically actually use exactly the same strategy that we would as chemists. And so bacteria use mixtures of N-formulated amino acids, or they use an N-formulated amino acid to initiate peptide synthesis. So as the peptide grows, it doesn't have this problem when it gets to the dipeptide stage. How does this relate to what we're doing? Well, I showed you that the products of this chemistry that we've developed, they're the N-formyl aminonitriles here. So what happens when you take these products and mix them with water or submit them to aqueous processing? There's basically 2 directions that this reaction can go in. And the reaction to the right is basically hydration of this nitrile group. And that's where you're going to eventually get to the amino acid, because you're converting this group over here to the acid group eventually via processing with water. On the left, you can remove that formyl group, and that sort of essentially undoes the difference between this and the Strecker chemistry, and you may as well be using Strecker chemistry. And the problem with what happens when you go over here is these compounds aren't stable. These compounds can dissociate. They can essentially lose ammonia. They can lose cyanide. These things can evaporate off, and you won't convert all of this material to the amino acid because they're fragile towards this Strecker reversion chemistry. So we tested out what happens when you take these compounds in water, and it turns out the reaction to the right dominates. And so what that means is when you take these formyl aminonitriles, you end up with the same sort of mixtures that I showed you are used in bacterial biosynthesis of peptides. So you end up with formulated amino acids, and you end up with unformulated amino acids. So again, this isn't a smoking gun, but it's maybe just a hint that this sort of chemistry might have been useful. It certainly doesn't solve the problem of primordial peptide synthesis, but it perhaps gives a protobiological system the available compounds to develop that solution by itself in terms of using a protecting group for peptide synthesis. Cool. So yeah, that's the same strategy that bacteria use to synthesize peptides. Cool. So that brings me to the end of my talk. So again, just to summarize, we're following the chemistry of this ever-expanding primordial soup. That's what prebiotic chemists are doing. We're developing or discovering reactions that we think are plausible and fit into these conditions, and that are fit into the geochemistry of the primordial earth as well. And as a result of that, we discovered these interesting compounds that we didn't expect, and also perhaps a solution or the means for a solution to be developed to the diketopiperazine problem in primordial peptide synthesis. So yeah, if you're interested in this work, we just published it a couple of months ago. So this is my growing group at the University of Otago, the iconic clock tower in the background there. So this is Sasha, she's an undergrad, and she was on this paper involved in this work, and also thanks to David Russell and John Sutherland out of MRC Laboratory of Molecular Biology in Cambridge as well, and to the funders that have supported my group, my new group. I've got scholarships and so forth available, so if people are interested, or if you know people that are interested in not only origins of life chemistry, but also biological and organic chemistry, please tell them to get in touch. And yeah, we can keep working on this really interesting problem towards the origin of life. Thanks very much.
Okay, thank you.
How many days of the year is it like that?
Every day, 365. Come join me. So that was in April. So, you know, that's not too bad, but I don't think it looks like that today. Yeah.
Hey, thanks for the nice talk. I was wondering, you know, formamide can actually decompose to CO and ammonia directly, just with, especially in the presence of a little bit of acid catalyst. I think you have formic acid in some of these mixtures.
Yeah.
And I was wondering, do you ever see just like the regular Strecker products from decomposition of formamide?
Yeah, so we definitely, when we do the reactions with ammonia, we definitely see those reactions. So I guess if you assume that the formamide could decompose and then you do the chemistry, I think you would expect that. When you start with fresh formamide, you don't really see that. You see like the N-formyl version of the Strecker reaction, which is kind of interesting, like, because you would expect it to be far less nucleophilic than ammonia.
Yeah.
But this reaction works. So yeah, we don't really see that happen.
Oh, okay.
So if you go back to the slide where you have the C double bond S compound right after the addition of the thiols and the
This one?
yeah, so you got, I mean, obviously on the left hand side, you got two electrical atoms, one is the double bond, the nitrogen is wrong. And so the first one reacts faster with the sulfur than the second. But now the product here, you've got two, but bio and a form, right? So both of those are hydrolyzed. And one of them, I guess, can hydrolyze by a cyclic. It's 500 grains of the sulfur attacking carbon.
Yeah.
Which one hydrolyzes faster?
So we haven't actually studied the hydrolysis of these compounds. So yeah, we were sort of content initially to say that all of these compounds really can act as precursors, or at least you would expect at least one of them for the different directions that they might go in terms of hydrolytic processing to act as a precursor to cysteine. But yeah, we definitely want to test the hydrolysis of these compounds. I think that the thioamides, they're just going to go to the carboxylates.
Yeah, I assume the SH system could deformulate it.
Yeah, but the key point is that at this stage, it doesn't really matter if it's deformulated in the context of amino acid synthesis, because you've locked in, so you've protected it from the Strecker reversion. Because you've hydrated the nitrile group.
Yeah, no, I got that.
Yeah. But yeah, this is underway at the moment. I want to see how these go in terms of hydrolysis.
Thanks, Nick. It's very cool. I was just wondering with the formulation that you described, you mentioned the bacteria to do this. I imagine the prebiotic system would be quite potentially catastrophic. And on one hand, that being organization, but on the other hand, how do you control formulation? You have too much of that, you could kill everything.
Yeah, it's a great question. So it's sort of a, it solves a problem on one hand, but it also blocks the reactivity of utilizing these in peptide synthesis. But the cool thing is, if you start from the primary products of our chemistry, it gives you mixtures of these, and the ultimate product from extended hydrolytic processing is that you lose the formyl group. So if you just fast forward into the future, this is an amino acid synthesis. Great, we've got another amino acid synthesis that's complementary to the Strecker synthesis. But if you take this at an intermediate point and we measure the hydrolysis rates, and of course that intermediacy will depend on temperature and time, etc., then you do get these mixtures, and you can sort of if you just use that time variable, you can dial up, this could be the major product at the beginning, fast forward a few years, it's actually the minor product. So we think that somewhere in that spectrum, there might be scope for biology to be able to develop that primordial peptide synthesis using those mixtures.
What's the extent of the didn't have that? Do you do just
The DKP problem is a very severe problem. Anytime you activate the end of a dipeptide, the major product is the DKP. So you're really fighting, like, you know, if you do, it's a concentration thing, right? When it's an intramolecular reaction, the effective concentration, those two ends are right next to each other. So the only way to solve it really is to develop, you know, synthetic chemistry, or to have really high concentrations, which are problematic in other respects as well. So it's a real problem. This isn't a solution per se, but it's maybe a hint that at least the inherent chemistry of these compounds maps onto biochemistry in a slight sense, and maybe there's something in that.
Okay. Yes, thanks, Dr. Lee.
Thank you very much.
Next, we have Jeremy, who is our next speaker. And Jeremy is a PhD student, worked with the Dr. Albert's group in UNSW Sydney. And Jeremy will today talk about the integrating ribonucleotide precursor synthesis with autocatalytic Formose reaction.
Very scary. All right. Thank you everyone for coming to my talk. As Soumya has very kindly introduced, I'm Jeremy of Quoc Tran. I'm a student at Dr. Albert Fehrenbach's group. And today I'll be talking to you about nonlinear dynamics meet biomolecule synthesis, or the other way I think about it is that in the context of what Jim and Professor Benner and Nick has been talking about selectivity and trying to prioritize that in the context of the earlier, this is what happens when we have a complex mixture and then we roll with it. So as Jim and Nick and Professor Benner has been introducing, the concept of a chemoton is kind of like a minimal unit of what a cell could have been like without all the actual identities of the molecules like protein, DNA, etc. And at the center or at the heart of the chemoton is the metabolic cycle that fuels the two other autocatalytic cycle, being template replication and membrane reproduction. Now, for people who are not familiar with the term autocatalysis or autocatalytic, you can think of it in an abstract way where you can have like A reacts with B, forming two copies of itself, or something like a positive feedback. And in a sense, life, which you have start with one cell and then you take in food and then it self-replicate, is an autocatalytic process. And the rest of the presentation will be focused on this metabolic cycle, or at least what it could be in the context of a prebiotic chemoton. So arguably one of the most famous forms of prebiotic autocatalysis is known as the Formose reaction, which Professor Benner has been doing a lot of work on. The reaction require relatively simple conditions. You need basic pH, really high temperature, and a small amount of autocatalyst or something to kickstart the reaction. And in essence, it converts formaldehyde, this one carbon simple compound, into sugars such as ribose. And to briefly go through the mechanism of the Formose reaction, you first start with this 2-carbon compound called glycolaldehyde, which Nick was talking about briefly. And then it integrates the formaldehyde to form this C3 sugar. And then that C3 sugar do a little isomerization and add another formaldehyde to form a C4. This C4 then breaks down into 2 of the copies of the original C2. So this is where the positive feedback or the autocatalysis really shines. And looking at this cycle, we ask ourselves, is it possible for the Formose reaction to serve as the metabolic cycle in the heart of the chemoton? And not just that, it also fuels into the template replication process. So that, just to reiterate, can the autocatalytic Formose reaction provide the building blocks for template replication? Now, a lot of people have done work on this kind of work on trying to make nucleotides, precursors, but we want to do it in a way that not just make the sugar building blocks separate from the nucleobases. We want to do them together. We want to integrate the sugar production and the nucleotide synthesis. So here we have the cycle that I've shown you before, but the trick is we tried at cyanamide. Cyanamide will react with glycolaldehyde to form this precursor called 2-amino oxazole. And this precursor has been, this pathway has been extended upon and worked on by the Sutherland group that Nick was part of. After that, the 2-amino oxazole can then react with the other members in this Formose cycle to form these intermediates, which eventually can lead to the nucleotides themselves. You have 3O nucleotide and ribonucleotide and their respective genetic polymers. So for people who are not familiar with the TNA or the serial nucleotide, you can basically compare the 2 structures here, where the structure are relatively the same, but on top you're doing it, making the nucleotide with a 4-carbon sugar, and here you make a 5-carbon. You're using a 5-carbon sugar, so you're simplifying the synthesis a little bit, make life a little bit easier for the prebiotic chemist. So we just run headfirst into the reaction by first mixing a little bit of formaldehyde, glycolaldehyde, and cyanamide and see what happens. Before we start heating the mixture, which will catalyze and kickstart the Formose reaction, we take a look at the mixture at 0 time heating. And then we see first, this is acetate, which we included here. So nothing weird there. And these 2 compounds are byproducts or side product of the Formose reaction that is outside the scope of this presentation. The interesting thing is we did not see a very significant or not at all a signal of glycolaldehyde. However, after heating it for 22 minutes, you can actually see the same product a little bit higher. But now you can see a very nice signal of glycolaldehyde there. And lo and behold, we actually get 2-amino oxazole, 2 nice peaks around here. So what's the next step? Well, the next step is to look at the next intermediate, the pentyl-amino oxazolene structure, which shown here. Now, NMR is very nice. It gives you the structure, but it's also not very sensitive when you're dealing with very minute concentration. And for that, we have to use a technique called LC-MS. Now we look for the way MS works is you actually look for masses. And this is the total signal for compounds with the same mass as pentylsaminoxazoline, including pentylsaminoxazoline. And then we take a little, like a slice of the signal, which gives us the spectrum. The spectrum shows the fragment of when you bombard the molecules with the same mass, and then break them apart. What fragments will you get? So you get that spectrum, and then we have standards that we made in the lab, so we know the exact structure of these standards, and you can actually see that they match up pretty nicely. So this supports our idea that these pentylsaminoxazoline could be formed during the Formose reaction. Now, happy with the idea that we can actually make these nucleotide precursors, we want to go further. We want to investigate how these things are actually formed throughout the Formose reaction, and not just say that they can be formed. But for us to do that, let's first take a look at what a normal or classic Formose reaction would look like. So here, first let's start with formaldehyde. At the start, you have like a period with very little activity or very little consumption. The reason is the members of this cycle have not accumulated that much, so the cycle doesn't turn very often. So as a result, formaldehyde is not very much consumed. But once these members actually accumulate, the depletion of formaldehyde happens very quickly, which is accompanied by the production of the sugars, including the members of the cycle. Once formaldehyde is depleted, however, as you can see here, we call this the yellowing point, because at this point, the mixture actually turns yellow. And this is kind of like known as the Tar paradox in Formose reaction. You can actually see that the production of the sugar stops, and that some of them decrease in concentration. The question is, will the production of the nucleotide precursor follow the same trajectory as the sugar production? So in order to track the concentration of the different species in the Formose reaction, we mix these compounds together in an initial mixture, and then we aliquot them into different tubes and then start heating them at the same time. And then after that, at every time point, we will take one tube out, put it on ice, stop the reaction, and then analyze it later using HPLC. So here we have two graphs, one that tracks the formaldehyde concentration on the left, the other one glycoldehyde on the right. At 0 millimolar cyanamide, which is the classic Formose reaction, you can see that the formaldehyde depletes relatively quickly, almost straight after it starts being heated, which is accompanied by a corresponding production of glycoldehyde. Now, if we actually increase the amount of cyanamide, you actually see the period of little activity, the lag phase, get extended and exponent, and it takes longer for the Formose reaction to reach this exponential phase. And the same can be said for even higher concentration of cyanamide. So the idea is that cyanamide doesn't just allow you to make nucleotide precursor, it also affects the kinetics of the Formose reaction. So this is one way you could modulate the Formose reaction in a prebiotic scenario. Now, interested in seeing how the concentration of cyanamide actually changes during this whole reaction, we repeated one of the set of the experiment, and then we try to track for cyanamide. Sure enough, you also see a period of little activity of cyanamide because the Formose reaction is not really happening. And then at the exponential phase of the Formose, you actually see that the consumption of cyanamide increases very rapidly until at this point where the Formose reaches the yellowing point, the concentration starts to plateau. This confirms that the consumption of cyanamide is intertwined with the Formose kinetics. And if this is how cyanamide looks, then what does that say about terminoxazole, the product of cyanamide and glycoaldehyde? Well, we do the same experiment, but track this with LCMS. But this time we try to make the reaction a little bit more fancy by introducing a 13C compound. The idea is that if you replace the formaldehyde with a 13C formaldehyde, after enough cycle, you actually get all the sugars made incorporating this 13C. And as a result, they will be heavier. And what that allows you to do is to distinguish whether your 2-amino-oxazole is being made by the glycoaldehyde you added at the start, or the glycoaldehyde actually produced by the Formose reaction. So first, we have this unlabeled glycoaldehyde 2-amino-oxazole, which even when you start mixing it at the beginning before heating, you can see like a small amount already, and that barely increased at all. But if you compare that to the 13C 2-amino-oxazole, you can see that it starts at 0. Production really ramps up at the exponential phase, and then you can start to see it's a little bit slower, but like the slope is not as steep as original. So this confirms that the production of this 2-amino-oxazole, a precursor of RNA and TNA, is fueled by the Formose reaction, and the kinetics of both processes are intertwined. Now we flip that around just as a way to get secondary confirmation. Now we have the glycoaldehyde having the 13C labeled and formaldehyde is the normal one. So now we'll expect the trends to reverse. And sure enough, that is what we observed, where the compound with the 13C doesn't increase very much or not at all. In some cases, even decrease, while the products that were made with the normal formaldehyde and the carbon 12 actually start at 0 and then picks up production during the exponential phase and plateaus afterwards. This actually happens for all the compounds that react between cyanamide and the Formose cycle intermediates. But here I'm highlighting the ones that are of our interest to nucleotide precursor synthesis. So here, as just to reiterate, 2-amino-oxazole serve as a common precursor for both these compounds. And then you have the TNA, which is the TNA precursor and the RNA precursor. Now, just before I reach the conclusion of my talk, I want to give everyone an overview by looking at the scheme. And so we start with this simplified Formose cycle. And I would like to use this opportunity to acknowledge that normally what you actually expect from the Formose cycle is what Professor Bennett showed yesterday, where there's a lot of complexity. But for our purposes, which is focused on the autocatalysis and the kinetics, we think that this cycle serves very well as a proxy. We add cyanamide, which gives us this little bit of a mess. But if you know where to look, you can see that cyanamide then forms a lot of these products that we don't really know what they're useful for, or maybe we just haven't found a use for them. But amongst this whole mess notwithstanding, you can actually see the precursors for the nucleotides, including the TNA, the terminoxazole, and the RNA precursor. And the thing is that as long as you still have formaldehyde in the system, the cycle can keep turning. And because it can keep turning, it can keep fueling the synthesis for these useful compounds. So just to wrap up with my conclusion, the autocatalytic Formose reaction could have fueled the prebiotic synthesis of nucleotide precursors as proposed by the chemotone model. two amino oxazole tetraols and pentols amino oxazoline, the DNA and RNA precursor have been detected, some of which has been confirmed with standards. The synthesis of these nucleotide precursors is dynamic, kinetically coupled with the kinetics of the Formose reaction, which in this case is mediated by cyanamide. And what we're trying, the message we're trying to push with this work is that we're trying to contribute to the experimental study that investigate the chemoton in the context of prebiotic chemistry, as well as trying to integrate the nonlinear dynamics, the exponential phase and the lag phase that comes with the Formose reaction with prebiotic synthesis. I would like to acknowledge Dr. Albert Franbach and our postdoc, Dr. Regent Yi, other members in the Franbach group, and UNSW and the Australian Center for Astrobiology. And thank you all for listening.
We have to wait for question and answer session.
So, like formaldehyde has, when it's in the presence of your mixture, it has two choices. It can react with base to EOIs and then go around the cycle, or it can react with cyanamide to form a product that you're interested in. So you have two microscopic rate constants. Okay, one of them is rate of realization, the other one is rate constant condition. So the rate constant realization presumably is pH of 7, I'm assuming it's increased pH by a factor, and by pH you increase the rate of realization by a factor of 10. The reaction rate constant for the addition of the cyanamide to is probably much more complicated than that, because you have to worry about acid base equilibrium or cyanamide itself. And actually, I don't happen to know the pK of cyanamide, whether it's a protein or a deprotein in the range of pH which you measure. But you think these concentration levels get 7 or more cyanamide do you actually have the rate constants for those two processes measured?
The short answer is no. The reason why we chose those concentration is actually because we actually test like a wider range of concentration at actually higher than 9 or like around 12. Cyanamide just consumes up all the members of the Formose plus glycoaldehyde, and it actually you get the same reaction as the Formose reaction without anything to kickstart. It just goes the lag phase and the Cannizzaro reaction. But at the small concentration at 0, 7, and 9, you can actually still, the two processes still compete with each other. And at the end of the day, the Formose autocatalysis wins out because it creates enough members of the cycle fast enough so that it doesn't get competed with the Cannizzaro.
But you must have a buffer measure. I think that 0.2, I get what that was, so it might drop that in that, didn't it?
Yeah.
But yeah, so 0.2 more. So you actually, I guess your buffer in this is the calcium hydroxide.
So in this case, we actually don't really have like the calcium acetate actually doesn't buffer very well. Yeah.
So the acetate is not buffer. Actually, it's buffer.
There's no buffer in the system. And actually, if you track the pH of the system over time, it actually decreases a little bit because of the Cannizzaro reaction.
Okay. Thank you. Thank you, Jimmy. Next, I'm very honored to invite Professor Rowena Ball from Australian National University.
Thank you. So it's a great pleasure to be here and reconnecting with chemistry and connecting with geology So I'm from ANU in Canberra, on the ancient and rightful lands of the Ngunnawal Nation. This is just an overview. This talk is just an overview of work carried out over the last about the last 8 years, off and on, with my partner in crime, Professor John Brindley at Leeds University, and where we hope it might be going. It's not going anywhere. No.
Use the mouse.
So if we exit full screen, then
Can you start the video?
No, I can do it.
so those are okay. Right. So broadly, we've been inching forward on 3 leading questions. The first concerns the far from equilibrium requirement. A system can be at a non-equilibrium, so a far from equilibrium steady state, or behave dynamically in a self-sustained way, such as cyclic or oscillatory behavior. We've been investigating synthetic systems driven dynamically rather than steady state. Secondly, given that the primordial prebiotic environment, wherever that was, we think would have been interfacial, non-uniform and messy, and subject to stochastic fluctuations, we ask, well, was a fluctuating environment necessary for complex prebiotic chemical systems to emerge and persist? And thirdly, how can we use the results of these studies to construct a probability landscape for the emergence of life, so that we could say with high confidence things like, for example, don't bother spending resources looking for life on this or that, perhaps, outer solar system moon. The probability is very low or very high. So various experiments have been done to support the hypothesis that a prebiotic system would have been periodically driven by thermal cycles. And this is one experiment from 2012 about replication in the primordial RNA world. So these experimenters use thermal cycling between 10 and 55 degrees centigrade to achieve replication of a 35 base pair piece made from a transfer RNA in a semi-batch system, and the fluorescence quenching arises as duplexes are formed. And they didn't get replication occurring under an isothermal. But in these experiments, the thermal cycling drive is externally imposed by the experimenters using a heating element. So we began in 2014, we looked at that and began by simulating this RNA system to replicate in a fully non-equilibrium flow-through cell in which the power input and thermal cycling are provided internally and self-consistently by a hydrogen peroxide thiosulfate redox oscillator with the reactants in continuous supply. So This is a thermochemical oscillator, well known since the 1970s. And in the cell, we get these uniform thermal oscillations between about 10 and 50 degrees centigrade with a period of a few minutes. So the enthalpy balance for our dynamical system is coupled to the concentrations of all species through the Arrhenius temperature dependence of the rate constants. And in a string of subsequent papers, we use this system to drive things like ribozyme action and dimerization, polymerization, a simple metabolism, vesicle formation. So that's all very well, but one thing is almost certain about the environment that hosted the emergence of life is that it was not like a laboratory environment. It was a fluctuating, messy environment. So to simulate these effects, we made the ambient temperature incident on the cell into a Gaussian quantity, fluctuating randomly about the mean with variance S. So the incident thermal fluctuations can also be treated as a proxy for fluctuations, for density fluctuations, or fluctuations of chemical potential. And I've drawn that little sort of normal distribution at the bottom to emphasize that, just to highlight that the input fluctuations are normally distributed. And we supply the hydrogen peroxide and thiosulfate as before, and we get thermal oscillations in the cell again, but they fluctuate in amplitude and period. We also supply the cell with various simple organics. They can be things like pyruvate, nucleic acid, and amino acid monomers, and simple amphiphiles. So not necessarily all at the same time, but in different experiments. And under the oscillating thermal drive, they form simple vesicles or micelles, small biopolymers, and feed a primitive metabolism. So the input fluctuation, as I said, fluctuations have a normal distribution. What does the fluctuation distribution of the thermal oscillation amplitude look like? Well, that's what it looks like. Normally distributed input fluctuations do not produce normally distributed output fluctuations. We get this left skewed, right weighted distribution. What does it mean? Well, we have this significant weight towards larger temperature fluctuations. And this means that on average, the system supports high activation energy synthetic reactions over the low activation energy degradation or reverse reactions. So we can begin to see how this non-equilibrium system with self-consistent fluctuating thermal oscillations favors structure, complexity, and persistence in the prebiotic world. So what happens if we suppress the thermal oscillations in the cell and run the fluctuating system at a steady state? Well, we get this left skewed, this right skewed, left weighted distribution, weighted towards small, low temperature fluctuations. And the large, high temperature fluctuations, which you need for high activation energy synthetic reactions, occur on average less frequently. So in this steady state scenario, over time, high activation energy synthetic reactions are going to occur less frequently than low activation energy hydrolyses and degradations. So the steady state scenario produces an anti-life distribution. And here we just compare the productivity of each of these fluctuating scenarios. So the steady state on the left and the dynamical oscillating state on the right. And these are both driving simple linked metabolic cycles that were studied experimentally in 2018, where the output is pyruvate and glyoxylate, where the input is pyruvate and glyoxylate and hydrogen peroxide, and with ammonium ions sprinkled in at the right place, out pops aspartate. And we found that the relative productivity, as measured by the production of aspartate in the system run dynamically, is 20 to 220 times that of the steady state system. Now, if we consider mineral surface prebiotic reactions in a non-equilibrium flow system, we have a situation very similar to what was described in the 1970s from this huge body of experimental research related to optimization of catalytic converters for motor vehicle exhausts. And that was all about oxidation of carbon monoxide on metal surfaces, mainly platinum. You have this surface thermochemical reciprocating system where there's this high energy adsorbed species that mediates oxidation of the surface site at low temperature. So you have on the low stroke this low activation energy exothermic reaction. Then the heat produced swings the cycle over to the endothermic high activation energy desorption stroke, which produces a temperature spike. But that cools things down. So the oxidation stroke takes over again. And so it goes on. So this reciprocating oscillator is always swinging to and fro for as long as the disequilibrium is maintained. It's just inexorable and generalizable to minerals in solution, and maybe a universal mechanism for driving prebiotic molecular processes under primordial conditions on rocky planets. So one review paper from that era put it this way: relaxation oscillations may be lurking in every heterogeneous catalytic system. One might speculate that every heterogeneous catalyzed reaction might show oscillations under the appropriate conditions. So let's look more closely at thermochemical relaxation oscillations. In the top figure, I've got this temperature time series of the relaxation oscillations produced by the hydrogen peroxide thiosulfate redox oscillator. And I've taken this piece between the two dotted lines and stretched it out to look at the temperature in the middle figure and rendered the piece in terms of the concentration in the lower figure. So for a long time between spikes, the temperature is barely rising. But you can see in the lower figure that the concentration, that the exothermic reaction is still happening. So where's the reaction heat going? Well, it's being stored as the potential energy of intermolecular vibrational modes. And it's only when that saturates that it's released as a temperature spike, released as kinetic energy. So this sort of oscillation is very typical of systems with high specific heat capacity. In other words, aqueous solutions with energetic surface active solutes, such as hydrogen peroxide. So these liquid systems with high specific heat can store a large amount of heat in the internal modes. And it makes you wonder, what the transient intermolecular vibrational mode populations might actually look like. Well, let's back up a bit and summarize. In what we've done so far, in prebiotic molecular reacting systems, self-consistently driven by a hydrogen peroxide redox oscillator, normally distributed input fluctuations produce a non-normal or skewed distribution of output fluctuations. This result implies that firstly, vibrational mode populations of the active medium are likely to manifest transiently as non-Boltzmann distributed. Secondly, on average, high activation energy synthesis reactions are favoured over low activation energy reverse and degradation reactions. And thirdly, that constraints are inherent on environments where it's possible for life to emerge and on probabilities of its doing so and persisting. So you wouldn't expect, in this scenario, you wouldn't expect to find life in like methane oceans of Titan because of the low specific heat and inability to sustain relaxation oscillations. So, because all of our published simulations effectively describe recipes for real experiments, I wrote all this up, as you do, I wrote all this up into an ARC Discovery Grant application. And this is what I wrote for Section A4, the 100-word non-specialist summary. And I won't read it out. And this is a lot of people here would have had to do this. This is the dreaded national interest test statement, this compulsory thing. I had to rewrite it. The research office made me rewrite it about 2,700 times before they were happy with it. And I was told it has to be written so the person in the street would understand what they personally might get out of the research. So I spun it in terms of molecular self-assembly. And eventually the research office was satisfied. Well, what's it all about? What needs to be done to get this show on the road? Firstly, we need FTIR validation of transient state non-Boltzmann populations. Now, the Boltzmann distribution is a bit of a sacred cow in chemical physics, but it was derived for and applies to dilute gases in equilibrium. Distributions different from that predicted by equilibrium statistical mechanics are in fact commonplace. And non-equilibrium statistics with stochastic dynamics may indeed exhibit non-Boltzmann statistics. So secondly, how exactly would a non-Boltzmann population distribution of internal quantized potential energy show up in the anomalous distribution of thermal fluctuations? Well, the simplest working hypothesis is that higher populated, more energetic modes dump more energy into higher energy temperature or translational fluctuations. And third, we need to carry out large-scale non-isothermal simulations of complex molecular systems subject to stochastic input perturbations in regolith pore geometry to obtain probabilistic answers to contingent questions, such as far from equilibrium is a necessary condition, but how far? And so on. Why might some extraterrestrial prebiotic systems fail to progress to life? And finally, build up probability landscapes for structured prebiotic molecular systems, which would enable us to select the most promising most probable systems to progress to fully fledged cellular life with a high degree of confidence. And finally, there's this compulsory section you have to address in a DP application with the heading cost effectiveness of the research and its value for money. So I pitched this comparison of emergence of life research with gravitational wave research in terms of these two fields having similar potential to be transformational in our understanding of life, the universe, and everything. So I'll leave it there. Thank you very much.
Any questions?
Hey, thanks for the nice talk. I'm wondering with the temperature oscillations, I understand how they could spike up very quickly, I guess, in water, but then why does it come down so quickly as well? I mean, the water would have to take a while to cool down, I would think. Is there an endothermic part of the reaction too?
Yeah, absolutely.
Oh, okay.
Yeah. Yeah, it's a exothermic, endothermic coupled system.
Yeah. Okay. Yeah. Very interesting.
We call it an index system, which is self-explanatory.
What's the rate of those thermal fluctuations? What's the time scale for those? Is it that like on milliseconds, or is it on days, or like what kind of rate?
Minutes. So depending on how you set it up, it can be yeah, a few minutes to a few tens of minutes.
Right. We have one more question.
Very interesting. And I'm not an expert in this field at all. So you did a normalized input distribution.
Mm.
If you can imagine a photon rotating, you can expect the temperature to go up and down on a daily cycle. Do you think that would affect your activation energies in any way? Or completely mixed up?
No, that's a very good question. We used and I don't know the answer. We simply used experimental activation energies or experimental Arrhenius parameters harvested from the literature for all the reactions in our system.
Okay, thank you, Professor Gold, for my time.
Yes. Which one are you using? Maybe this one? Yes. So there we go.
This is the point. Yes. Next, we have Shotaro Tagawa from Tokyo Institute of Technology and Earth and Life Science in Japan. And today he will talk about deep sea supercritical CO2 water environment type prebiotic nucleosides of phosphorylation. So, what do you?
Thank you for giving me a chance to sharing my work. Hi, I'm Shotaro Tagawa, a PhD student at LC Tokyo Tech from Japan. Today, sorry, I mistaken the title. Today I will talk about prebiotic nucleoside phosphorylation under deep sea supercritical CO2 and water two-phase environment. Prebiotic phosphorylation reaction leading to prebiotic nucleoside phosphorylation leading to prebiotic nucleoside phosphorylation has been so far reported from the condition the condition simulating wet dry cycle near terrestrial hot springs with atmosphere and water. In many cases, the condition involved urea and cyanogen as a catalyst or reactant. The advantage of having an atmosphere is that water can evaporate from the aqueous phase, which results in the condensation of various organic molecules. On the other hand, in the deep sea, which is one of the plausible birthplace of life, it is considered that molecules will undergo hydrolysis due to the presence of abundant water. In the past 20 years, natural CO2 fluid has been discovered in deep sea hydrothermal fields, such as Okinawa Trough and Mariana Arc, like these bubbles. The model shows that a subsurface pool of liquid and supercritical CO2 exists. So far, supercritical CO2 is mainly used in the industry to as a green solvent to dissolve various hydrophobic molecules like caffeine. Hence, recently proposed liquid and supercritical CO2 hypothesis assumes that two phases of SiCO2 and water drives prebiotic chemical evolution near the deep sea hydrothermal system. An aqueous phase adjacent to the CO2 fluid is assumed to be highly acidic and thus dissolves various ions such as phosphate from apatite minerals. In such environment, phosphate will be accessible and further undergo dehydration and condensation to drive phosphorylation. So we experimentally tested such possibility. We used nucleoside as a target for phosphorylation. Aside from its importance in biology, using general analytical instruments such as phosphorylation, such as synthesis via phosphorylation reaction in the SiCO2 and the water two-phase environment, where water is released from the water droplet and the reaction solution is concentrated. We have developed a hydrothermal reactor system using an autoclave to achieve the phosphorylation reaction in the SiCO2 and the water system. First, nucleoside was mixed with phosphate source and other chemical reagents. Then the solution, the reaction solution was added to inner cylinder, and then the solution was flash frozen using liquid nitrogen, and then liquid CO2 was injected on top. Finally, reactor was heated by oil bath and under the different temperature and pressure and duration as shown here. After experiment, the sample was subjected to UPLC and LC-TOF-MS analysis. First, we investigated nucleoside phosphorylation using uridine nucleoside and sodium dihydrogen phosphate and urea. Reaction condition is 70 degrees Celsius, 10 to 12 megapascal, and the 24 hours. This is the result. We observed uridine phosphorylation under the SiCO2 environment only, and with urea greatly enhancing the reaction. I showed the chromatography and MS spectrum, which suggest that several isomers of origin monophosphate were identified only in the presence of yes, SCO2 and yes. About negative control of SCO2, we checked the yield of UMPs in the water-rich environment using this gold tube, and then UMPs weren't identified in the negative control. Next, we performed a nucleoside phosphorylation using hydroxyapatite as the only source of phosphate. As a result, we observed the similar yield UMP isomers compared to the experiment using sodium dihydrogen as a phosphate source, which suggests that phosphate is indeed released from apatite and further used for the phosphorylation reaction. It has been predicted that pH of an aqueous solution coexisting with CO2 fluid become highly acidic, and at this acidic pH range, hydroxyapatite is known to dissolve in water. And next, I investigated temperature dependency of nucleoside phosphorylation. Yes, origin phosphorylation proceeded over 60 degrees Celsius in the SCO2 condition, and the yield of UMPs can be higher with increasing the temperature. Increasing temperature is likely to cause UMPs to decompose, but perhaps pressure suppress this tendency. Yes. Next, we investigated the comparative time course experiment using 4 different nucleosides to understand the difference in their reactivity as well as duration duration needed for the reaction to reach equilibrium. Yes, UMP has shown its maximum yield reaching 17%, reaching over 70% after 120 hours, and followed by CMP and GMP in that order. Whereas the yield of AMPs was the lowest in nucleotides after 120 hours. The overall trend suggests that the reaction rate differs among nucleosides, and perhaps due to the difference of pKa and or the reactivity with urea, etc. Finally, I will talk about urea function for this reaction. Previously, the two different reaction mechanism has been proposed. First, a model by Sutherland proposed that urea is directly reacting with phosphate to form reactive urea phosphate intermediate to be attacked from the nucleoside hydroxyl group to form a nucleotide. Whereas the Katsura and Inagaki proposed that urea first decomposed into isocyanate and ammonia, which in which in which isocyanate further react with phosphate to yield carbomoyl phosphate, which against react with nucleoside to form nucleotide. Yes, we challenge to detect the origin phosphate intermediate as well as carbomoyl phosphate using phosphorus NMR, but we could not observe the signal indicating such compound. So we went back to the UPLC data, and then we found the mysterious unknown peak, which did not overlap with non-standard chemical compounds. Interestingly, LCMS analysis revealed that this compound has M over Z of 288.1, which is equivalent to the mass of carbomoyl nucleoside. So now we are investigating the possibility that this compound could be the key intermediate to be phosphorylated. This is summary. We confirmed that two-phase SCO2 and water condition can drive phosphorylation reaction using hydroxyapatite, and increasing temperature can enhance nucleotide yields. And yes, time course experiment has shown the difference of reaction rate and final yield among nucleosides. Last, we found carbomoyl nucleoside intermediate is formed during the reaction process, which may lead to a new reaction model. I especially thanks Associate Professor Fushima and the three students who mainly support my research. Thank you for listening. ありがとうございます。
Any questions?
Yeah, thanks for the very nice talk. I was wondering if you noticed decomposition of the uridine at all.
Composition.
Decomposition, maybe loss of the nucleobase, something like that.
Maybe very small. Maybe this very small peak is equal to uracil, so nuclear base. But yes, but very the peak is very small.
Yeah. And where do the reagents and products actually prefer to be? In which phase? Is it the water phase or the?
Yeah, I'm curious about that, but my reactor is I couldn't develop the method to get sample from CO2 phase and water phase.
Yeah.
So now I joined JAMSEC, and then in the institute I will try to make the sampling system.
Oh, yeah. Right, like a window or something in the reactor, so you could shine a light through and detect that way. Or...
Sorry, one more.
Oh, we can talk later after.
Okay, thank you.
Other questions?
Look, I have a naive question of biologists, so I'm not sure if I understand everything that you set up, but carbon What sort of effect is in your transitional temperatures? What is the effective pressure on molecules like that?
Stable. Yes. If in my opinion, the it's sorry, out of range. High temperature and high temperature and pressure maybe if high pressure in the high pressure condition CO2 and water phase I think mixing below so I think the effect of water the effect of water for decomposing some compound is decreased and yes and sorry I couldn't answer. Sorry.
Do you think it's more stable in your experimental setup and it's low pressure, high temperature?
Low pressure.
So if you just heat up in one atmosphere to 100 degrees, it's carbon monoxide, great, right? But in your experiments at high pressure, then degradation is much slower.
I predicted so.
Okay, thank you, sir, for nice presentation. This is the end of the session. Now we have a break and then we'll start it. Thank you.
And so we're a little bit over time, but if everybody wants to come back at 10:55, we'll get started with the next session. Hey. You want to use your own laptop?
Let's go to OneDrive. Is that all right?
My account.
I do, but I don't want to get out of my
that make sense?
That's why you're getting a portion of it, so it's not like getting one, like getting the particular stuff.
This is one excited state in H atoms. And we're deliberately targeting. So this is actually 121.56 nanometers, is that famous Lyman alpha emission. Right, except rather than going this way, we're going that way. Right, because once we have things that are in, we've already given it 10.2 electron volts or so, it's pretty much easy to do anything with the atom after that. So all we're doing is just getting the electron from n=1 to n=2.
And so I think my follow-up question was, is that like a singlet state, or is there like also association the pyramid state, but
That's one electron system. Sorry.
Alrighty, can everyone join me in thanking Chris for a wonderful talk? Next up, we have Dr. Soumya Kanti B from UNSW, who will be talking to us about carbon dots.
Hello, everyone. I'm Soumya. I'm a postdoc in AAM2023 lab. And today my topic of discussion is carbon dots and unexplored multifunctional prebiotic material. So basically, I will talk about the prebiotic availability of the carbon dots, which is a carbon-based nanomaterial, and how they can affect in different process of life if they existed in the early environment. So first thing first, what is carbon dots? So carbon dots are basically carbon-based nanomaterial, have a size in the range of 1 to 10 nanometers. And they generally primarily consist of sp3 hybridized carbon with small amount of nitrogen and oxygen from their starting material. They have unique optical and electronic property, which is very different from their bulk material, and they produce distinct emission behavior depending on the size and the surface defect of the particle. So in short, carbon dots are carbon-based fluorescent nanomaterial. So how we synthesize carbon dots in now? So generally the synthesis procedure of the carbon dots is very easy. All you need two key ingredients. First, you need a carbon source and you need a source of heat. So what people do, people take a source of carbon and they put it in a desired solvent, for example, water, ethanol, methanol, etc. And they either put it in an autoclave at optimum temperature from 100 degree to 200 degrees Celsius, or sometimes they put it in a micro oven. And depending on the time, they can produce distinct carbon. And you may ask, what is the source of carbon? So people use different kind of carbon sources. Nowadays, people use honey, orange juice, lemon pea, coffee ground, wood products, and even people use cow dung as a source of carbon. So anything that contains carbon can be converted to carbon dots with proper environment, temperature, and the process. So this is the general introductions about the carbon dots. Now we hypothesize that these carbon dots can be possible in the prebiotic environment, and why we hypothesize that we'll discuss in the next slide. So in origin of life research, we often said that in order to understand how life originated, we must ask what were the chemical or present. And we also believe we need to also ask what other new materials are present in the environment and how they can affect in different processes of life. The introduction of a new material, one that we have not yet thought of as a prebiotically available, could potentially have significant impact in the origin of life research and indeed in the emergence of life. If a new material exists, whatever it does, for example, it may accelerate some of the process or maybe inhibit some of the process, but whatever it does, we need to consider this material in the environment. And that is why it is important to know what other new materials are present in that environment. As I work in the nanomaterial field, I observed that although our human body or life cells are full of nanomachines, for example, proteins, enzymes, people rarely talk about nanoparticles when they talk about how life started. So, and we all know that carbon is heavily involved in the life and in terms of organic molecules, biomolecules, but its nanomaterial form is not even considered yet. So we hypothesize that carbon nanomaterials are present. And this hypothesis comes from a very well-known reactions, which is Fischer structure reaction conditions. Fischer structure reaction condition is a series of chemical reactions that actually converted carbon monoxide and hydrogen into defined organic molecules. So people use mainly oxalic acids and put it in an autoclave, and depending on the temperature, actually they converted into they degrade into carbon, mainly degrade into carbon monoxide and hydrogen. Then they produce different type of organic molecules, for example, fatty acids, fatty alcohol, and etc. etc. In prebiotic scenario, it is very illogical to think that a molecule which is formed in a high temperature and pressure, if after synthesis of that molecule, they immediately transport to an environment where the pressure and temperature is optimal. So a molecule which is synthesized in the high temperature and pressures, that will be in the high temperature and pressure for the longer period of time. So we need to ask what happened to those molecules if we again react by the Fischer structure reaction conditions. And that particular conditions is very much feasible for the carbon dot formations. And that is why we hypothesize that if geological environment would have supported the Fischer structure reaction conditions, then it can also support the carbon dot formation reactions. Now, how can we get the carbon dots in prebiotic scenario? Again, we need two key ingredient things. First, we need a source of carbon. We have a prebiotic source of organic molecules in the prebiotic scenario. They may synthesize in the early earth, or they may come from asteroid impact. And we have a lot of different type of organic molecules with the carbon is the main constituted compound. In that particular thing, I wanted to mention 3 organic molecules like citric acids, some amino acids, and sugar molecules. These 3 molecules are extensively used by the researchers to produce carbon nanodots or carbon dots. So we can also use those, prebiotic chemistry also use those organic molecules to synthesize the carbon dots in prebiotic settings. Second thing, we need a prebiotic heat source. We know that terrestrial hot spring have a up to temperature 290 to 120 degrees Celsius. Some hot spring have a temperature more than this thing. Also, lava flood have a temperature greater than 200 degrees Celsius, and sometimes the water stream mixtures can reach very high temperature. Apart from the hydrothermal synthesis, there are also various procedures people use to synthesize carbon dots, for example, thermal decompositions, pyrolysis, carbonizations, and UV assisted synthesis. If you look at these procedures, all these procedures are natural occurring procedures and may have been existed in the early earth. So we believe that carbon dots may be possible. Carbon molecules can use any of these synthesis procedures and may be converted into carbon dots. So we wanted to synthesize carbon dots in the prebiotic scenario, and we choose a simple method, which is thermal decomposition, to synthesize the carbon dots. And for this reason, we need a source of carbon. And we choose fatty acids because we know if there is a protocell, there is a good possibility to fatty acid in the environment. And we also know that in fatty acids, short chain fatty acids are more prebiotically available than long chain fatty acids. That is why we choose decanoic acid as a source of carbon. And what we do, we put it in a porcelain crucible and put it in a furnace for 2.5 hours and change the temperature from 110 to 190 degrees Celsius. So initially the decanoic acid is a white, it doesn't have any color. And when we change the temperature, we saw that at high temperature, it produced a defined brown color liquid, and we extract that liquid with ethanol extraction method and put it in a UV lab. So we all know that decanoic acid is not a fluorescent molecule. It doesn't contain any fluorescence. However, when we put that extracted compound, we observe that the high temperature, it produces a fluorescence emission at 150 to 190 degrees Celsius very strongly. Then we studied its fluorescence emission. We observed that fatty acids or decanoic acid does not have any excitation and emission, but the product we extract by ethanol, it's have excitations at 340 to 350 nanometer and have emissions around 460 nanometer. And it gives us an excitation dependent emission. That means when we excite in defined region, it gives an emission at defined region. And the excitation dependent emission is a characteristic behavior of the carbon dots. So we thought that it may be a carbon dot. Also, there is a lot of characterization techniques still needs to be done. For example, HR, chem analysis, XPS, etc. But this is the preliminary result. Then we try to synthesize vesicles from these carbon dots by using decanoic acids. So if you remember the synthesis procedure, we use decanoic acids to extract the decanoic acid carbon dots. And then we thought if geological environments support the formation of carbon dioxide from decanoic acids, there will be free monomer of the decanoic acid, and therefore we add decanoic acid and then we make a thin film and hide it in the buffer and wanted to see what happens. And we observe that there is a formation of vesicles, and we still try to understand whether they are vesicles or oil droplets. But the interesting thing is that although we did not add any fluorescent molecule to tag those vesicles, all these vesicles are oil droplet fluorescence by nature. And I believe that if those things are carbon dot, the vesicles also have that property of this carbon dots. Now, how the carbon dots can affect in different process of life if they exist in the early environment? We hypothesize that it can affect the compartmentalizations by affecting growth, division, and the nutrition uptake. It also have a very good catalysis activity. Therefore, it can affect the metabolism, and it can also act as a UV protectant in that environment. So first, how they can affect the compartmentalizations. So imagine a thin film of the lipid and the carbon dioxide. Now, if you hydrate that thin film, you will find out that defined model of the nanoparticle lipid interactions. It may absorb on the surface of the bilayer, maybe incorporate in the bilayer, or maybe in the core. But whatever the interaction model is, it interacts, the carbon dots will interact with the bilayer. And when it interacts with the bilayer, they actually alter the membrane packing. When they alter the membrane packing, they actually alter the membrane phase state. So we know that the alteration of the membrane phase state or growth and division is really dependent on the alteration of the membrane phase state. So when they alter the membrane phase state, it's possibility that they can also affect the growth and division of the vesicle. The growth and division of the vesicles can be more clearly visible in my next slide. For example, imagine a two scenario when there is no carbon dots and there is a with carbon dots. So without carbon dots, lipid vesicle can grow in surface area, but needed to have a strong curvature to induce division. Such curvature can be achieved when we incorporate the carbon dots in the bilayer. Second, we know that normally the bilayers are mainly impermeable. But when nanoparticle interacts with the bilayer, it is known that they produce the pore formation, and this pore can be further used for the possible nutrition uptake and the release mechanism. It can also affect the metabolism. It is like a carbon dots, catalytic activity of the carbon dots are used by the chemistry people extensively. They used in oxidation reduction, and recently it has been also shown that they can convert the carbon dioxide into several organic molecules. So that particular property can be also useful in the prebiotic scenario. We also know that we have a strong UV radiations, so protecting the organic molecules from the UV radiations in one of the reasons that we will propose the life in the hydrothermal vent. So it can also absorb the UV light and can protect the organic molecules from the photo damage. In this section, I wanted to show a particular literature. This work done by the Phuong et al. And they published in 2019. So they produce a carbon dots which may not be periodically available, but they mix up with the amino acids and they show the formation of dipeptides, peptides, and the protein molecules in presence of carbon dots. And what they show that most of the amino acids, this is the conversion of the amino acid to dipeptide. So this shows that the most of the amino acids actually converted to the dipeptide form, and even at -20°C temperature, the yield is around 20%, and when the temperature is 25 degrees, the yield is around 80%. And we also know that our prebiotic scenario may have more than higher temperature than 25°C. So in a conclusion, there is a possibility to develop carbon dots or carbon nanoparticles in the prebiotic conditions. And if they exist, they can affect in a lot of different processes. For example, they can affect the growth and divisions, they can affect the permeability, they also can affect the metabolism, and also can protect the different organic molecules from the photo damage. So I want to thank you, my lab mates, and particularly Dr. Hansen for support. I want to thank UNSW Sydney and Human Frontier Science Program for fellowship, and AM committee to give me an opportunity to talk. Thank you.
Yeah, copy that.
On slide 12, we had 3 Now, can you slide what that the question, but how would the availability of other elements inhibit or induce carbon dioxide formation? I mean, your experiments, you using particulars of trying to picture in the geological environment.
Normally the carbon dioxide can be produced, anything that contain carbon can be converted into carbon dioxide. For example, if we have one source, if we have two, 3 or 4 source, all of these things can be converted into carbon. For example, you have 4 different type of organic molecules, they will all be converted into different carbon dots in proper temperature or proper pressure. And now the main question is that, like, other materials, how they can affect. For example, we know that the metals ions affect the formation of the carbon dots, maybe sometimes they precipitate it, but there are also reports that when people synthesize the carbon dot in the presence of defined metal ion, it actually doped inside the carbon dot and it actually enhanced the property of the carbon dots for catalytic activity and etc.
Thanks for the fascinating talk. Could you talk a bit about what you think the fate of these carbon dots might be geologically in a pre-life Earth? So if they're formed in, say, a hot spring pool or a lake or an ocean, what is their geological preservation ability? How would we recognize it?
Recognition of the carbon dot is really a little difficult because first thing, they have a distinct fluorescence emission property, but that will not work because of the presence of other molecules. It may quench the fluorescence intensity. And another way to really understand how the carbon dot is synthesized or not, we perform the HRTM images, but I don't know how I can prove in the geological settings to whether there is a carbon dot or not. And I'm still trying to find out that thing. It's difficult because it's really small. It has a size around, mainly average size less than 5 nanometer, and it's difficult to detect in terms of other molecules.
So yeah, can I just clarify on the synthesis? Is it using Fischer-Tropsch chemistry to get to the fatty acids, and then is it a cross-linking, like oxidative cross-linking process to get to the carbon dots?
Carbon dots can produce both in oxidative environment and reducing environment. So I am just saying, like, if a molecule is formed in high temperature and pressure, that will be in the high temperature and pressure for longer period of time. And anything that contain carbon, whether if we use the autoclave reactions, or we can also use several other methods, for example, thermal decompositions, for example, pyrolysis, carbonizations, or UV assisted synthesis procedure. There are a lot of procedures available to convert carbon molecules into carbon dots. Even, like, simple organic molecules for formaldehyde can convert it into carbon dot. We will use formaldehyde in the autoclave and they produce carbon dots at a little bit high temperature.
So in Fischer-Tropsch chemistry, they directly to carbon dots.
And yes, but like, if you put organic molecules, like for example fatty acids in autoclave reactions, after a day you can find carbon dots, but I have no idea, like, if we see from the starting of the reactions and what happened after several days.
Thanks.
Yeah, last question.
Thank you for the talk on slide 17, where you showed the showing them ice formation. So just one question. Working condition, just they make use of carbon dot and that's it. And what are the source of that carbon?
Oh, it's I think it's something graphite. They use arc discharge method to produce that carbon dots. I don't know whether it is periodically possible or not, but yeah, they just use carbon dots and visible light and just solution of amino acids. No.
Okay, please join me in thanking Soumya for your talk. And we will now be breaking for lunch. Thanks, everyone. We'll be back here at 1 sharp. Thank you.
Very good.
You're on a place.
Yeah.
Yeah.
Hey everyone, welcome back to the final afternoon of AAM. Thank you all for engagement so far. We have 5 talks left, and so we have 2 talks, a quick coffee break, and we'll keep things on time because some people have to get on a plane ASAP. So first up, we have an invited speaker, Professor Penny King, who was one of our brave poster judges yesterday. So thank you for doing that. Yeah, and she will be telling us cool stuff for the next half an hour.
Okay.
5-minute warning.
Can you give me a 10-minute warning?
10-minute warning.
Thanks. Lovely to see you all, and thank you for the invitation to be here. Today, I'm going to tell you about some of our adventures with gas-solid reactions. Now, if you're a geologist, you probably never heard of a gas-solid reaction. I did hear the phrase earlier from the chemists, but geologists actually don't talk too much about gas-solid reactions. They're only just making it into the textbooks, but we know that gases and solids exist and coexist, in fact, in this room. So I think they're a potentially important place to be looking at reactivity on other planets as well. So this is work done with quite a large group, and it's ongoing. Oops, I'm going to get this to work. There we go. So as you know, Mars is a very dry and cold place. It does not have as much atmosphere as Earth. It's not well shielded from radiation, and yet here we are looking for life on Mars. The surface is also not particularly encouraging for life, containing a lot of sulfates, chlorine-bearing minerals, and very few carbonates. Yet still, we search for water, because water is important for cell function. And water is an essential component of life. And so the Mars exploration program has built itself on this once tenet, following the water. A tenet that actually I'd like to note goes back tens of thousands of years with many Indigenous people, including the First Nations, people from Western Australia, where some of you are catching the plane. Where the Wandjina spirits, and I just love these pictures, this rock art, were the cloud and rain spirits that brought water to the people, creating the landscape and its inhabitants and allowing life to thrive. So we look for water and we try to find it. Here's an example of a paper showing geomorphic evidence for water on Mars, and we find it in Mars's early history, which is important because that is when we think life arose on Earth, and by extrapolation, potentially on Mars also. So water here is shown in a variety of forms from the geomorphology, has then been sort of mapped out in this wonderful review paper, and seems to cover a lot of these areas where there is rock rather than dust covering the surface of Mars. That story has also been put together with a story of early volcanism on the surface of the planet. And so we've heard from people about how important volcanoes may be, life springs, and the origins of life. And here is a diagram from a recent paper showing how water might be sourced at volcanoes, transported into those valley networks that I just showed you, entrapped in the crust as hydrated materials or perhaps frozen. And so then that builds a history combined with the minerals that we can detect from space of entrapping clays through volcanism and sedimentation early in Mars's history. A middle period with potentially more neutral or acidic situations with sulfates, and a drying of that climate with eventual oxides at the surface. It's a lovely story. Can it be that simple? Well, I was wondering that and thought, well, actually, we could go back to fundamentals of chemistry to think about that. and try and pair chemistry with geologic processes. And so here's a very simple diagram of a cross-section through the crust of Mars, showing the different types of geologic processes. So when we've been searching for water on Mars, we've been focusing primarily on these blue sedimentary hydrologic processes, those beautiful rivers and craters, things that Joe's talked about too. Joe's also worked a lot on the volcanism and the transfer of volatiles from the deep mantle to the surface. There's loss of that material to space, thinning the atmosphere, photochemical reactions, and of course, impacts in the early history of Mars. So what, in a fundamental chemical way, does that mean? Well, it means that we have liquids and solids in the case of the sedimentary processes. And then we have melts, solids and gases, or melts, solids and gases in the two magmatic and impact processes. So we spent a lot of time, and I've even worked in this area looking at liquids and solids, but what about those solids and gases? Is there a role for high temperature gas-solid reactions? And that's what I've been exploring the last maybe 8 years. So what is a gas-solid reaction? Well, you know about these, they happen in the fridge. Ice goes to gas in your freezer or back again, vaporization and condensation. We have gas uptake reactions, and here's one of Andy's beautiful pictures of a micrometeorite coming in and reacting with the atmosphere, so becoming oxidized. And we also have so-called surface-mediated chemisorption reactions. So this is where we have a gas, like a volcanic gas, reacts with the surface to create new materials and usually a new gas. So this is all described in this book, Blatant Advertising, and the main takeaway points of this book are that these reactions redistribute chemical elements and they transfer electrons. Very important for building those cells and for transport of material. They're controlled by kinetics, very important from the perspective of what Rowena was talking about this morning. So they're disequilibrium reactions. We can't use thermodynamics very well to predict them, and they occur over seconds to minutes, very fast. They make soluble salt products, so some of those things that are on the surface of Mars, like halides and sulfates. We investigate these. As I said, not much has been done. One way to investigate them is to take a material like a basaltic glass, supersaturate it with volatiles that when you heat it, they're expelled into a closed silica tube. And so the gas then travels from a high temperature down the to colder ends of the tube to deposit phases. So this is work with Hannah Neckvissel's group at SUNY Stony Brook, and they've tried to simulate Martian gases that you would get out of a volcano, and you precipitate minerals like those we have found in Martian meteorites and on the surface of Mars. Another way to do this is to actually run your gas through a furnace instead. And so this is work we've been doing at AAM, where we flow gas into a tube. Here's our tube again, over different solids, and then have the gas go out. And what we find when we do this, as I mentioned, is we have reactions in seconds to minutes. And what happens is you end up growing new products on the surfaces of these materials. Here's some of our results of putting different substrates into an instrument with SO2. We didn't add water because that makes sulfuric acid and suddenly you don't have a furnace anymore. But I'll talk more about water and how we've got around that later. When you put a pyroxene into this kind of situation, it rapidly forms sulfates and iron oxides on its surface. Olivine less so. Isn't it interesting that there's so much olivine on the surface of Mars? it's always puzzled me that so much olivine on the surface of Mars that it doesn't react very well with this kind of situation. We've put glasses in, and excuse the garish colours, blue is for calcium sulfate, green for magnesium sulfate, and we end up with the glass turning into this amorphous material as it reacts underneath. We can write reactions for this kind of process. We're making sulfates. We're making amorphous material that's rich in silica and aluminum, like we see on the surface of Mars. In fact, up to 30% of the material the Curiosity rover has looked at has been amorphous. We also make reduced sulfur in the gas phase. If we look in cross-section of what this might look like, we end up with our alteration minerals on the surface, sulfur dioxide going in, a reduced sulfur going out, iron oxides, and an amorphous layer that's just on the surface of the material that forms a barrier to continue the reaction, perhaps contributing to that 30% of amorphous phases on Mars, and an important sort of mineral for remote sensing, because remote sensing picks up molecular bonds vibrating, it doesn't pick up actual minerals. So is this really true? We can do this in the lab, right? Is it relevant to Mars? Well, the results are actually consistent with thermodynamic models, which doesn't mean much. It just means that we've, tweaked our thermodynamic models properly to choose the right phases. But we can predict with a theory what might happen. We can't say whether it will happen. So what we thought at that point was, well, okay, that's good. We can predict. Can we predict for Mars? Well, maybe we should go look at Earth. So Earth analogues are what we decided to look at next. And here is nicely degassing volcano at Kilauea. These volcanoes degas over tens of thousands of years and release low density gases into the atmosphere. It's hard to avoid water though. Well, that's okay. This kind of collaborates that evidence of lots of clays and hydrated oxides on the surface of Mars. So that's fine. But what if we did try and avoid water at a volcano? What would we see? Well, I was lucky enough to be in Hawaii in 2018 when it was erupting and able to collect some of the ash. So here's a cartoon of Kilauea, and you can see the magma chamber going to the surface. We could sample from the plume, and the ash in the plume is reacting with that very hot, greater than 800 degree mixture of water, SO2, and hydrogen chloride species. And this is what we find. We find glass rimmed by these reaction rims, very similar to what we found in the lab. So anhydrite in purple on the outside of these rims with that amorphous type material here in this case ends up being albite, a sodium anticlase mineral, and amorphous silica, and sulfur in the reduced form in the gas. We also can find material from the lava lake, richer in fluorine. And so these are fumarolic materials or lava lake materials, and we can find materials from the hydrothermal systems that have been sampled by these ashes. That's great. So now we can use this data to test thermodynamic models. So we looked at the phases present, we put in our phases, we tweak around with the temperatures, and we can then actually duplicate the phases with a thermodynamic model. So this then gives us confidence of using those thermodynamic models on Mars. So we did that next. Took our average Gusev basalt, potentially an average basalt on Mars, added volcanic gas to it. So used a typical terrestrial volcanic gas because we don't know any better, although a Martian gas potentially has a bit more chlorine and sulfur. And we can then thermodynamically model what you might get. So here's a plot of temperature versus abundance. And we make a lot of calcium sulfate or anhydrite. as the most abundant of the calcium phases. If we look at the iron phases, we can look at a range of different gas to rock ratios. And if we don't add much gas, we make a lot of sulfides. If we add more gas, we make more oxides. And so we can tweak our situation dependent on what we want to make or what we want to, if we tweak our situation, we can produce different things. And I think this helps us explain why we see a lot of iron oxides on the surface of Mars and very few iron sulfides, is that we don't actually have that much gas. We can also look at what you need to do to make clay, and you can see that you need to get to quite low temperatures, and of course helps if you have a lot of gas. What does this do to the atmosphere though? That was our next question. Well, if we look at the gas that's coming out, we can see that sulfur dioxide takes a huge plunge. That kind of makes sense. We don't have much sulfur dioxide in the Martian atmosphere either, or in the Earth atmosphere, because it gets taken up in sulfides and other minerals. The water is also lost as you make the clays, and the CO2 is lost, but it's the highest remaining gas remaining in the atmosphere. And in fact, I think this could be explaining why CO2 is the richest gas in most other planetary atmospheres. So let's sort of summarize where we've got to here. So we have these volcanoes, they're erupting, they have a mixture of gases and solid particles and melt coming out of them. We're taking that material, we're reacting it, we are creating surfaces that are picked up by remote sensing that are rich in these secondary minerals that are found and abundant on the surface of Mars. And that process is actually diluting the gas and producing water that's then episodically available to make our beautiful valley networks and so forth, and then eventually ice, since the orbit of Mars is such that water's not going to remain stable for long. So what about impacts? Impacts are also going on and also able to create these gas-solid reactions. We had a beautiful talk from Gretchen showing us the crater abundance on Mars, and we can use that dating technique that Gretchen presented to show that there are abundant Noachian impacts. And in fact, some authors have suggested that between 46 and 100% of the surface of Mars has actually been resurfaced by impact processes. I think that's astounding. Each impact involved heating, some of it not very hot heating, but there's substantial evidence, and I could go into that later if you want, to show that the larger impacts are sufficiently large to vaporize those salt minerals that are on the surface, creating gases and solids in the expanding plume, which I'm trying to get my arrow to, but it's not really working. On the right-hand side. So there's going to be a trajectory of temperature, a temperature gradient within that plume where there's an opportunity for gases to react with solids. And remember, this can occur on a scale of seconds to minutes. So what are we impacting into? Well, I think that the Noachian probably looked a little bit different than the current Mars, but we know that there's a lot of dust and volcanic material, shown here on the left. And we know that in the present day, that a lot of the hydrogen is now locked in mineral spores in ice in the poles. And so there's a possibility of thinking about, well, if you're impacting into that, we've had these volcanoes going off, making altered sediment. And we also have ice and water that's trapped. So those are potential ingredients that we might want to look at for a model here. So what we've done is taken the average of all of that dust, so the average Martian soil, and looked looked at that and said, okay, let's add in different amounts of water, pretending as though you might be impacting into the surface of the planet with more or less amounts of ice or entrapped water. So that gives us models that are slightly different than the magmatic degassing one, because we're starting with a different starting composition. We're starting with the altered material. And so here's a plot for the sulfates that are produced. And again, anhydrite is the most abundant of the sulfate materials that forms. If we look at the iron minerals, iron oxide dominates by far, and we don't get sulfide anymore. The sulfur in this case goes into the gas phase. And clays are going to be dependent, thank you, to a large extent on how much water you're impacting into. So obviously, if you're going to impact into more ice, you'll end up with more clay material because there's more water in your bulk composition. The carbon, interestingly, stays in the atmosphere. Carbon's quite a linear molecule, so chemists will know this, and it doesn't have a strong dipole like sulfur dioxide does. And so the carbon actually finds it very hard to react with the solids. And so there's not much carbon sequestration out of the Martian atmosphere, so the carbon stays in the gas, whereas the sulfur dioxide does not. It's trapped and toxic and corrosive and makes those sulfates. Okay, so let's summarize this kind of pictorial model. We have our volcanoes going off, taking that volcanic gas, reacting it, condensing materials in that sublimation process, so the gas going to a solid. Also, the gases reacting with the surfaces of the materials, making new solids, resulting in something that looks like this. Altered amorphous volcanic sediment that from space looks like a bunch of olivine because it doesn't react very much. Looks like a bunch of calcium poor pyroxene because the calcium has gone into the anhydrite and we end up with pigeonite on the surface of Mars, and a whole lot of diluted gas. We also have impact going on, and I like to think of that impact process as kind of like a scrubber. Think of your dryer, and you're taking that volcanic material, putting your gas into it and tossing it around. And you're doing that repeatedly with impact over time. And it's taking that water and trapping it into minerals, taking the ice, trapping it in minerals, and also into the sulfates and chlorides. So that process is occurring repeatedly, building up all of that dust that we see on the surface, creating massive amounts of sediment that are then able to be transported around the surface of the planet to build up thick sedimentary sequences that we see and are really puzzled by on the surface of Mars. And taking that material, trapping it in minerals, and also freezing it at poles where there's then limited volatile transfer between the poles over time. And so we then have a more diluted atmosphere in the end. Okay, so hopefully the end of all of that, I know I've taken you on a bit of a wild trip through a whole lot of different techniques and processes, but hopefully I've shown that we've been able to do experiments, use field data, and I didn't show too much of the thermodynamic modeling because it's a bit boring, to be honest, to show that basalt reactions can produce the alterating materials that we see on the surface. Thermodynamic models can constrain those reactions, tell us where we're going, not necessarily how fast. The repeated impact processing can accentuate that process and also produce the iron oxides that we see more extensively than iron sulfides in the surface, and that these processes draw sulfur dioxide and water out of the atmosphere, creating the thin CO2 atmosphere that we see today. So, any questions?
So when the exoplanets that they're starting to look at, you can only see the atmosphere, you're never going to see the surface.
Yeah.
Realistically. I like your final conclusion there, and I wondered if you might, is that a general thing that you can figure out what's happening with the solids of the surface by looking at the atmosphere? Is that a general result there?
So I've actually had a student, Sarah McIntyre, we were trying to look at that. And then we realized all of a sudden, we actually don't know the pressure on this and the temperature on the surface of these planets, these exoplanets very well. We better figure that one out first. So we did that calculation and figured that one out first, which was published earlier this year. And then we're moving on to the second one. Now, the thing about exoplanets is the exoplanet systems actually have a different bulk composition in some cases because the stars have a different bulk composition. So what we might model for our solar system is actually different than what you might model for an exoplanet solar system. And we started off by modeling using our sun and the appropriate bulk compositions there and what might you then produce. And we're now at the point of, well, we better look at the other star compositions, and it turns out they're quite hard to find. And so the elements that we want are quite hard to find.
Very excellent.
So yeah, we are moving that way. Thank you. I mean, it is the next obvious thing, right?
Well, and I think
And we're looking at Venus as well,
yeah,
which is also the next obvious thing. Yeah.
yeah, the hot ones will be the next down one.
Yeah.
It's easier.
Yeah, we just wanted to make sure we actually knew the pressure, surface pressure and temperature first before we went too far. Yeah. Yeah, Andy.
Have you guys looked at the beauty of the pressure measure and what do you think of the old pressure?
Yeah, I'm getting a piece from Carl actually, so I will be. Yeah, I can tell you that. So here's the pyroxene composition for the meteorites. So you would think that if you do have this impact scrubbing, that you should be able to see it through the Martian crust in the meteorites. And so this is showing the crustal rocks and their calcium. So calcium on these diagrams for people who are geologists is up the top. And these contours refer to the temperatures that these minerals crystallize at. And the bottom line here is that as you go from deeper in the crust to shallower in the crust, this field is kind of moving down to less calcium in these minerals that are at the bottom. So this is Mars rover, and this is regolith breccia pyroxenes in some of the upper class. So yes, I think that Black Beauty is showing this from the data that's here, but I'd like to collect a bit more.
Ask you a question, otherwise we can catch Penny during the coffee break. Yeah?
Yep.
All right, so
Thank you.
Penny.
Share my screen. Yes, sure.
So next up we have Dr. Joshua Hedgepeth. Yeah, who is also an invited speaker, and we're just going to switch over to his laptop right now.
How do I need to share on Zoom or not?
Yeah.
Yep. So it's disabled.
Oh, I gotta let you in.
So you recently moved here from the US?
Yeah, well, actually, technically I was in Canada for 6 years before I.
Oh, okay, sorry, not North America. Yes.
Yeah, still, yeah, same continent.
Same continent, yeah.
Okay.
Okay, great.
Okay. Thank you. Actually said, I am Joshua Hedgebeth. I recently started a postdoc at Curtin University working with Katerina Milchkovic, as well as her other postdoc, Rowena Jones, who presented on similar work on Tuesday. My work is supposed to be building on that, sort of the modeling side of the remote sensing work she's doing. And what we're doing is we're investigating hydrothermal conditions and impact craters and how that might have implications for habitability on Mars or in the subsurface now and past. And I'm going to be doing some basic reviews. I want to create a story here before I get to the work that I actually did. I mean, as astrobiologists, it's obvious why we're interested in Mars for life, but it's not just us who have an interest. I mean, all of the entire planet has been fascinated with Mars since before we even started the space race. But for the better part of 3/4 of a century, sorry, I'm talking very fast. The better part of 3/4 of a century, we've been sending missions, even after they failed one after the other, to Mars. And there's got to be a reason for that. Why are we so fascinated with this planet? Perhaps it's because it's our closest sister planet. Maybe it's because it is the most Earth-like, with a terrestrial surface and similar interior, and some evidence of liquid water in the distant past, whether that water was present as an ocean world, like what we have on Earth, or in the form of subsurface lakes beneath ice, I think it's a little debated to some extent, but the takeaway is clear. It has similar compositional resources for life and liquid water, and of course, various energy sources, meaning it's very interesting astrobiologically, as many people have discussed throughout the entire meeting. Of course, that period of most habitable environment was quickly degraded away as we lost the atmosphere. As has been discussed already, the Neogean ended during the Earth's Archean, so that basically right as life started to arise, the ability for life to thrive on the surface of Mars went away. When we think about the long-term evolution of the habitable environments on Earth, we have to think about then the trajectories life might have taken. And what we're looking at here is just a schematic of certain scenarios that may have taken place on Mars. We can imagine habitable environments pictured here in blue compared to the uninhabitable version in red, and how that evolves through time along the x-axis. And what you can see is that there was most likely habitable environments on the surface and into the subsurface, like here on Earth. Over time, that likely evolved. That region, if it's still there, would have constricted, likely made its way to the subsurface. You know, that might have been there would either have to adapt and migrate, or it would have been made extinct. But what this really shows us is that today, if we want good evidence to try to investigate what life might be, it's in that subsurface. We have to understand that environment and whether or not it is still conducive, or whether we can find evidence of life there. And of course, that means we need to understand the water content. What we're looking at here is just a near surface water ice content across the surface of the equatorial region of Mars, that is to say up to about 60 degrees in either direction. And this is, just like I said, subsurface. It shows that there is water there. But when we think about long-term habitability, we want liquid water, and we also want probably a thicker layer, something like, say, a cryosphere. And the exact estimation of what a cryosphere or even groundwater perhaps will look like is, I think, still being worked on. There are some estimates that I will touch on later in my talk and its implications for my work. But the point is that if life is going to thrive, it's going to be in this region down here where water is available. If you keep going down deep enough, as the geothermal energy increases, you may even reach a region here, where you might reach hydrothermal conditions. That is important. Hydrothermal conditions, which are important because it's not just water and potentially resources. You start to have energy to facilitate chemical reactions and as sort of an environment for life to arise or perhaps thrive. And it's in those regions where I want to start to focus, the regions where we have evidence of liquid water and water processes. And I want to focus on that as sort of an indicator for habitability to some extent, and try to understand how we can use these various water-based minerals across the surface to try and understand those subsurface conditions, and how we can tie the mineralization that is occurring in water to the environment that would have been fostered in these various instances. Specifically, I want to focus on many of the things that are existing during the period where there would have been water, just as in the Neogean, which is where a lot of the deposits are in this most ancient part of the surface in red. If you look at the type of mineral deposit that's associated with, that these are associated with, rather, the geologic features, the highest fraction is going to be the impact craters. So a lot of the mineral deposits that we're seeing are being either created by or somehow associated with those impact craters. And the question then becomes, if there seems to be a large relationship to the craters, what can that tell us? Well, we already know that when water interacts with a hot source, a heat source, it can allow certain reactions to occur. That is what is allowing these minerals to occur. The type of minerals that actually form are going to be unique to the temperatures that are at play. And the beauty of an impact crater is that when it forms, it creates this beautiful temperature anomaly that, if large enough, could potentially intersect that cryosphere. It could allow for hydrothermal processes to occur, and if we could constrain that temperature anomaly, maybe we could start to make some predictions of the type of minerals that would be able to form in that thermal anomaly. But there's an added complication here that we don't necessarily know they came from that thermal anomaly. It's possible they form in the subsurface and they make their way through these fractures and faulting that occurs in the bedrock, the surface, to produce those examples that we see. But we can't really differentiate at the moment between those type of mineral deposits and those that would have been excavated by the impact process. Now, most craters aren't going to be able to excavate down to the depths that we hypothesize the cryosphere or groundwater would be stable. But as I've already shown, there is at least some limited ice content, or maybe ancient material could have been buried at some point, meaning there's just other ways in which these minerals might make their way to the surface, whether it might be in the subsurface to be excavated by impact craters. So we have to then begin to try and frame this question in a way that allows us to differentiate between what is being sourced from the impact crater process, that is to say, formed in the impact crater, or simply being exposed and made observable by the impact process. So that leads me to my major goals here, or major primary objective in this work. We want to differentiate between what is being sourced and what's being created. That's important for us to, one, understand which impact craters are most conducive to hydrothermal systems, but also as far as when targeting these regions as indicators of habitable environment. If we want to go there to look for more specific evidence of life beyond just these habitable indicators, we need to be able to make sure that we're going to is actually a sample related to hydrothermal activity in impact crater, as opposed to some other type of old process or tangential process. To do this, we have three basic approaches. We're going to start by identifying where the minerals are able to form. That's as simple as looking at the thermal anomaly that's created by the impact, and then saying, in this region, where are these different minerals that we'd expect to form in hydrothermal situations on Mars? Where could they form? And does that intersect with where we expect the cryosphere to be? Or will it be able to migrate? Is there a pathway within the fractures we'll be able to predict to make it to the surface? Which I guess leads me to number two, which is we want to be able to identify specifically where these pathways, type of faults that we hypothesize would be. And can we then make predictions on the direction or the pathway in which the location in or around the crater we might expect to see any minerals from the crater or specific minerals depending on what's being formed in thermal anomaly. And then finally, we want to use this information to constrain the diameters where thermal anomalies are likely to intersect with the cryosphere. And of course, it's going to be dependent on existing constraints, but it still allows us to sort of begin to say, if this is not a viable candidate, if the thermal anomaly is not deep enough that it will reach the depths at which we expect the cryosphere, then anything we see at the surface almost certainly must have occurred from excavation rather than being sourced from the crater itself. So, with that in mind, we're going to start with Tora crater as an example. Looking at here is just two different perspectives of the rim on the right and a spectral imagery in the middle, just to sort of highlight some of the variability that's going on there. Arita showed in her talk some of the mineral signatures that we see around the rim, largely focused there. But the reason I'm focusing on Tora crater because it's about 40 kilometers, about midway in the path of the range of complex craters that exist on Mars, which I didn't mention before, is the focus of our work. Because if we want to talk about a thermal anomaly that's large enough to reach the cryosphere or groundwater, we're talking about some of the larger craters, like 20 kilometers larger. And this we thought was to be a good starting case, not just for investigation, but also to create sort of a framework for us to model this. The model that we're using is called ICIL. It's a hydrophysics impact modeling code. which is basically able to simulate what would happen if a certain impactor hit the surface. It can create a thermal anomaly. It can also make predictions on the damage and pressures and type of strain that will be imparted on the surface, which will allow us to begin to extract information about the fracture or faulting system that can be created. And then from there, we could begin to make analyses about this. The problem with this model, with any model really, is the parameter space. We have to make sure that the parameters we're using to recreate Toro crater is actually reliable, as if they're one consistent what we expect on Mars, and two recreating Toro as we actually see it on the surface. To do that, that means modeling Toro crater specifically and extensively in a range of different scenarios. One of that scenario might be at different velocities, from the lowest one we'd expect being 6 kilometers per second to the highest 12 kilometers per second, and then at a range of different bedrock conditions. The simplest approach would be just no porosity, assume a constant bedrock and move forward with that. Otherwise, we can consider some constant porosity, above non-zero, that is to say. In this situation, it might be a bit truer to life near the surface, true to life in a geological sense, near the surface. But the most realistic, of course, would be exponential decay in pressure as you go down. And the reason we're doing this, as opposed to just going straight to the most complex, the goal is to keep the modeling as simple as possible without having to add added complexities to the approach. The main takeaway here, we start to compare this computer, the results to actual reality, is that there doesn't seem to be a lot of variance. First thing I'll say here, I'm only showing at 9 kilometers per second because what the takeaway is the same. The velocities don't seem to add that much variance, and it is part of the image. What you see here is consistent with what you see with the velocity changes. The more porous material will tend to be more compactful, have less rebound, as opposed to the non-porous blue line here will experience more rebound as it's less compaction able to go on. The other thing to consider here is, well, one, the DTM profile I'm showing you here is an average of the entire 2D profile on Quoc crater, which means these peaks and valleys are being sort of averaged and dampened down. And it's actually going to be not just that averaging, but it's going to be variation where the peaks are along the crater, meaning we don't necessarily use this line as a most true life picture. It's just enough to show that comparatively it seems to be fairly consistent as far as size and brim position. And not to mention, on top of that, rather, the middle portion here, or the rebound, is often considered to be some of the less reliable parts of the ISO model because the boundary conditions can lead to a bit excess in rebound or compaction due to how it feeds into that boundary condition. But the takeaway is still this. Every scenario seems to produce a crater that is effectively within the deviation that we see of this DTM, or this profile around the crater itself, around Toro itself and the life. So with that said, we can consider the nose porosity, the simplest approach, as a safe bet to move forward in our modeling for other craters and for Toro itself. So first takeaway, looking again at that temperature model that I showed before, we then ask ourselves, what region or will particular minerals be able to form if water is present? That's a key caveat there. And what you're seeing here on the right is basically these 6 categories of hydrothermal possibly formable minerals. And the domain in which they're sort of colored here is saying at this temperature range, this is the temperature range at which these can form. It's important to note that this thermal anomaly is actually intersecting the geothermal anomaly, where if you go deep enough, you'll be at natural temperatures for these things to form. Whether or not there was actually groundwater or cryosphere down there is a big question. And once you go over 100 Celsius, what will happen down there is a curious question. But this is going to have, I think, some important takeaways as far as when we think about the fracturing system. And when the fracturing actually digs into the geothermally, naturally hydrothermal region, what will that mean about what we're seeing on the surface? That's a question I'm not prepared to answer quite yet. But the main takeaway here is we can begin to see how we can visualize the region in which particular minerals may form. The hottest ones being, of course, to the hottest part of the thermal anomaly. We start to think about the fracturing that's going on. What we're doing here is considering some of the parameters that ISO is able to track. Damage being, I think, the easiest one to sort of interpret. which is itself a sort of a lasting impact of the total plastic strain through time, and then volumetric strain just being a measurement or metric for the strain in the system. And really, if you look closely, you can see the same sort of fractures or lines forming. It's important to note that this is sort of preliminary interpretation because we need to make sure that the damage that we're seeing actually will relate to a fault or a fracture, as opposed to just say increased porosity in that region, or if there is a fault or something going on there, are these going to be stable? Is that big enough depths, or those are big enough craters and deep enough depths, they might begin to get impacted. And so even if there is a pathway at one point, how long will it actually stay there? That's an important caveat that we have to investigate further. I also want to note that we're probably going to see the time step for these three is a bit early on compared to the final crater. I chose to show this because it's more true to life than the final product. There's a limitation in ISL. These properties are very sensitive to the evolution of the model. Now, as you get rebounds, that can cause excess damage to occur and give the appearance of more fracturing. So if anything, what you're seeing here is a conservative estimate of the type of fractures we'd see. And what we want is a pathway for stuff to reach the surface. This gives us a constraint to start to work with. And if we think about how they're intersecting, at least for Toro, it looks like it's going about, I believe this is, what was it? I think one radii depth deep. So we're talking about the fractures going down about 1 radii in depth and about 1 radii, I guess that's actually 2 radii out from the actual center of the crater. Meaning if we assume this type of same pattern in different craters, sizes, different diameters, we can sort of begin to estimate the extent to which these fracture systems will occur. The exact fracturing approach of each individual crater will be investigated further moving forward, but for now, we're focusing on Toro. Another thing I meant to mention, I think, was that the thermal anomaly itself, you can see that it extends about 50 kilometers down here, which is about 3/4 of the actual radius. So the depth of the thermal anomaly appears to be about 3/4 of that radius, or rather of that of the radius. That's correct. When we think about exposed versus formed, we look here, like I mentioned before, when you get to larger craters, it begins to intersect this natural region. We aren't prepared to say exactly what that implications are for our results. What's enough to recognize is that it appears that there should be pathways for each of the minerals to reach the surface. The question then becomes, though, is would you expect the cryosphere to intersect with all three of these? Maybe it will intersect with the spectite region, but not with the hottest up here, the green, it's called the framework silicates. And those are kind of predictions we can make depending on to what depths the thermal anomalies extend. And it's also worth noting when we get to the cryosphere, the groundwater system, the predictions, there's a lot of variability, both spatially and temporally in time. So if we can have constraints on the time at which they are formed, and of course the location, that allows us to make predictions on what minerals should be sourced from that crater, depending on the size of the crater. The actual extent to which the depths are occurring now, we might expect between 4 to 7 kilometers in depth, where we might actually have access to water, meaning our thermal anomaly needs to extend down to or beyond maybe 7 kilometers to be most conservative. But of course, the extremes are higher, but in the ancient past, it might have been closer to 2 kilometers. It's the most liberal estimation here. And we go and consider then all possible diameters, where I know the X and Y axes aren't that easy to see. Every crater is proportioned to be the same scale, rather, on the screen, even though, of course, it's increasing in size. The takeaway is still the same, about 3/4 of the radius in depth is how far the thermal anomaly will go. And as we get to bigger craters, you can see the possibility of geothermal activity somehow influencing our results or influencing what we see at the surface begins to get larger and larger. But there's still that caveat of having to understand the fracture system and the stability of it at ever increasing pressures. I suspect we may be getting the ability to source that deep warm region, there's probably going to be a point at which those fractures just won't be stable. For the most conservative cryosphere estimates we have here, I think it's about 6 kilometers in depth for a 20-kilometer crater, meaning for more conservative cryosphere depths, we may not even be able to see any minerals being formed in that type of crater. The same is true as if it's down to 4 kilometers, you wouldn't be able to see above smectite. So you wouldn't expect the crater to form things like the carbonates or any of the higher temperature types like things. So again, given the specific minerals we're looking at, the time period we consider the crater to have formed, and the location, we can begin to make predictions and compare that to observations. And in a way, this could feed into our estimates of the actual cryosphere. Of course, there's some circular reasoning that has to go in there, but there's still ample room to begin to constrain whether or not what we're seeing at specific craters are being sourced by the thermal anomaly that crater forms. So ongoing work, as I've hinted at about this, fracture analysis is a big one. We need to understand the size, porosity, and the stability, that is to say, if they collapse. And then the temporal evolution and possible fluid flow, where if we can model the fractures quantitatively, we might then consider a thermal or temperature evolution model, and maybe fluid flow, something like hydrotherm, which has been done on Earth and to its lesser extent on Mars, to consider actual direction of where material would go in these different systems. And of course, comparing with observations is the big takeaway, how you can test these results and without a consistent reality. And this here is just showing some of the initial analysis coming from Arita from her presentation on Tuesday. And so the takeaway, we've used the iCell mission iCell model to model different impact craters and to begin to estimate thermal anomalies and fracture profiles or other fracture system that would be created from this process. And we tried to tie that to the actual cryosphere or predictions of the cryosphere to make some predictions like, say, it appears to be that if we're going to have a crater that won't show any evidence of that isn't going to create a hydrothermal processes in the crater, it's probably going to be 20 kilometers or smaller. But even then, that could be changed given different caveats, because there's going to be variability with crater size, crater age, and crater location. But that also, again, means we have ample room to sort of test different or control different variables in this approach. And of course, lastly, is the different future work we can do. Thank you so much for your time. I look forward to your questions.
I'll start with questions, I guess. I noticed so your model does capture some ejection of material, but that's very small. So is that a crazy high resolution thing you just showed us?
So we are doing high enough resolution, so we should be able to identify the ejecta, and it's something we're going to investigate before we publish this work. We didn't include it here because we realized the extent of our model was basically cutting off the ejecta, so we couldn't get a clear picture. We're actually currently running models as I speak that will get a high resolution look at the ejecta, because that's honestly very valuable. Obviously, we have that should have a defined temperature expectation in that region, and if there's water at the surface, some source there, that means we can predict which minerals would form there. And that's, we don't need a fracture pathway to reach there. It's a direct prediction of minerals that could be plausibly found there. And I think it's outside of one radii, which is where the fracture system appears to be concentrated.
Right.
Thank you.
Other questions for Doctor?
Yeah.
I was just wondering, with regards to the compared to more market or your models. Like if you're reaching that wave, maybe getting more water. Does that change the fact that you're using the model?
So that's a good question, because as we interact with the cryosphere region, the way in which that will influence the actual crater formation, I think it's more complicated. If there's ice there, I don't think that will have significant impact on how the morphology of the crater. If there's water, actual liquid water that is, that could affect the morphology of the crater on the surface and the subsurface in a way that I'm not prepared to predict quite yet. And perhaps for us to really understand the fracture system as we get deeper down, we might need to start considering the effect of water on the material properties, which really complicates the problem, to be honest.
So how big a rock do I have to drop onto Mars to get access to so if I put deceleration engines on Phobos, slow it right out of orbit and drop it onto the surface, is that big or small with respect to the crater that it would put to get down to where the life is subsurface?
I think it's big. I don't remember what were the size of the projectiles. I want to say it was on the order of tens of kilometers for the biggest ones that I used to get an 90-kilometer crater. I could find that easily enough afterwards if you want those details, but for a 40-kilometer crater. I think that was maybe 1 or 2 kilometers in diameter for that projectile. So I guess it gives you an idea of how we're already sort of interacting with that over 100, near 100 Celsius domain where the smectites might plausibly form if it was water there. Yep.
So Sudbury has a very lot of things on sheets. Well, across. Like a 200.
Mhm.
Much bigger. So that has a minimal area underneath. Could you possibly add in something like a milk sheet?
Yeah.
Minimal area with.
So that's a good question, or a good point rather. The fact that we have a fracture system doesn't preclude the fact that there will be a melt pool that will likely cake that. I guess we are expecting some level of fracturing in that after it cools to allow things to be exposed. We can possibly make predictions if it will make its way towards the surface, whether or not it actually gets there is still a big if. There's a lot of unknowns, I guess, in that process. All we could do is, I guess, begin to hint at what's plausible if it can make it there at all. I think that's a good way to really test, to consider the effect of a melt sheet on the fracture system is a good way to really test the basic assumption at all that they need it's possible for anything to reach the surface.
One final question? Okay. Otherwise, please chat to Josh and Penny during the tea break. And let's thank Josh one more time.
Thank you for your time.
So we will do that to the philosophy where it's a little bit longer and start.
Still talk faster than I go to that. Thank you much. Oh, 4 years to
So it's just. Now, look. Have you heard okay. That's great.
Creators.
Are you making a joke about that?
Yeah, I'll.
All your secrets will be out. Okay, let's do that.
Okay.
All right. Hey, everyone. Welcome back. We have 3 final talks. They're going to be great. The first one is an invited talk by one of the ACA's newest additions, which is Dr. Indrani Mukherjee. So we're really excited that she joined us here and she'll tell us all about best science.
Thanks very much for inviting me. My lord, I've never been accused of being quiet. Yeah, so hopefully everyone is very well caffeinated because yeah, I've got some exciting stuff for you. It's very exciting for me, but I'm not sure how it's going to come across to you all. So I'll start with acknowledging Australia's First Nations people. I will take this opportunity to remind everyone what an important year this is for Australia as a country. And I've been engaging myself in finding more about the Voice to referendum, not for or against, just gathering more information. And I think it's beautiful. I think Penny mentioned it in her presentation as well, that we kind of share a similar history with First Nations people. They were one of the first scientists. They've been here for 65,000 years. I'm sure they would have also pondered about the natural environment around them for them to survive that long as much as we are, or we have been finding out or trying to find out the origins of life and what it all means. So I'd like to acknowledge that before we move on. I didn't have the next slide in plan, but after attending the last 3 days of this conference, I think it's been quite an experience for all of us here. I mean, if Jim's in the audience here, I think every time I eat cream caramel, I will always remember what caramelization of sugar means from now on, and your boron and dancing molecules of Phuong, and I don't know, it's just been quite mind-boggling, to be honest with you. And this phrase here, or the saying that Stephen Hawking said, don't read it because I'd like to read it out for you. The human race is just a chemical scum on a moderate-sized planet orbiting around a very average star in the outer suburb of one among 100 billion galaxies, and that we're so insignificant. And I've felt that in the last 3 days, honestly. I'm more of a scum than you all, I think, because some of the stuff that were being discussed at this conference or meeting has just been quite mind-boggling. And I was thinking, especially, you know, if you were to look at my face today during Nick Green's presentation, this is what I was like. I mean, can you imagine Larry David at this meeting and we're talking about all the reactions of what, I don't know, caused his existence on the first place? I mean, it's pretty mind-boggling, and it is so because we still can't agree on what life is. We've had all this conversation for the last 3 days, and if we were to ask, all right, everybody, we've had our time, we've given our presentations, right, can we then define life and recreate it in a lab? I don't think it's going to be possible, but I leave it all to you because thank God it's not my field of expertise. And I'm going to leave it up to you all, incredible brains in the room that we have here today, because I'm on the SEAL side here. Honestly, I have no idea what's going on. But what I am interested in is to find out fingerprints of ancient life. I know we've been floating on Mars, Venus, and whatnot, but I'd like you all to just come back to Earth for a quick second, because tell you what, I actually don't know still that if we have a robust way of telling what's life on Earth and what's not. And when it's a fossil or an organism that's running around like the cute little dog in that presentation, remember, it's pretty self-explanatory. But when something is a biological organism and it's being replaced by a mineral completely, is there a way that we can say that there was once a life form and now it's not? And Nick, if you will notice, this picture is the only common thing in both of our presentations. I am not going to show any high-level chemistry as you did, but this has been the trajectory of my research, really. I work on the chemical or the mineral pyrite extensively for all sorts of reasons, but recently
not recently, but we all know that pyritized fossils or microfossils are quite common. So here's a very good example of three-dimensional preservation of cellular material. So when it's that straightforward, there's no question of whether it's a life form or not. For instance, this one here is we've got subcellular preservation in giant ostracode sperm. Like, this is exceptional preservation in the rock record. This is not what I'm talking about when we're trying to look at what are the signs of life. This is very obvious that it was once a life form. I'm more interested in when something is completely replaced by another mineral. And in my case, because I've worked extensively on the mineral pyrite for other research topics, I've been quite keen to see, well, we're forming pyrite in the water column in our sediments all the time, but pyrite is also one of the common minerals that replaces a biological organism. Can I tell the difference between what's biological or not? And if tomorrow Joe Michalski finds pyritic fossils in the lake sediments on Mars or wherever, and he brings it to me and say, can you tell me if there was a life form there? Then hopefully I will be able to help him, I don't know, 100 years from now. So installation in progress, probably not a good idea. And here's the next click problem now. So there are two types of mineralization that we're interested in. One is biologically controlled mineralization versus biologically induced mineralization. And there's a difference between the two. And most geologists will agree the control and the induced side will have a fight in literature, forever. Oh, that wasn't a biologically controlled mineralization, it's an induced one, and vice versa. I'm not interested in that fight. As long as it's biologically related is something that I'm interested in. So just very quickly and I've given you examples of both of these here, that biologically controlled mineralization is when the organism is producing that mineral because it serves a physiological purpose within the organism. So here we're seeing some a magnetovibrio organism, and we're forming these magnetite crystals, as we can see, and it aligns itself as well. Biologically induced mineralization, this just depends on the reactions of the cell waste products of an organism, and whatever is on the exterior of the organism, it reacts with that and just forms a magnetite on the exterior of the cell. So this is magnetite. This is magnetite as well, and they are formed by different processes, but regardless, they're biologically related. Yes, the mechanism of formation it might be different, and the reason might be different, but it's still biologically precipitated. So there are these two distinctions that I thought I'd mention. And the other difference that needs to be highlighted here is the difference between permineralization versus orthogenic mineralization. So these are the two differences here. So here we're seeing example of permineralization, a thin section through a fossilized stem. And here we're seeing an example of orthogenic mineralization. In this case, it's completely pyritized ammonite. And so what's happening in the permineralization is that the replacement of the biological organism or cell structure, whatever is being transformed, is because of an infiltration and permeation by mineral charged water. In this case, however, in case of orthogenic mineralization, it's a product of activity of decay bacteria. And I know it might sound counterintuitive that if I'm saying something is being replaced by another mineral, why is it decaying? Decay is very important. As long as the rate of decay does not exceed replacement, that's fine. But you need the decay of that organic material to provide the initial sort of reactants for the mineralization to occur. So there are these two different kinds of processes, and I will focus more on the orthogenic mineralization side of things. So here's an example of petrified wood. Just quickly going through some of the, and so this picture here, or reflected light photograph of wood, is from the late Devonian, from the Nick deposit in Canada. And so when there's any biological material, in this case, it's a plant one, so, it depends how well the material is replaced by another mineral, depends on how much decay has occurred. Now, remember, the decay cannot exceed the rate of precipitation. Of course, the nature of microbial activity, because these decay processes are highly regulated by microbial activity, and different microbial activity will have a different set of reactions, element requirements, etc. What mineral forming ions are present in the near vicinity? So of course, in our case, we would definitely need iron and sulfur and all those things, and trace elements, hopefully, for me to understand what's biological and what's not. And of course, whether the precipitation is induced microbially, so by that microbial community. Sometimes it's the, pretty much autolysification of the microbes itself. So some of the microbes are formed of apatite, they lithify and they form the orthogenic mineralization themselves by their own lithification. The unit size of the crystal that's precipitating, unfortunately for me, the smallest size of the pyrite crystal is not small enough to capture every single detail of the organism that's being replaced. So it really depends on what it is being replaced by. And then, of course, taxonomic controls. Are the soft parts of the organism being replaced, or are the hard parts being replaced? And so there's a whole range of things that matter for when this pyritization occurs in a sample. In my case, it's pyritization, but it could also be replaced by, for instance, apatite. So I showed you an example of ostracods before, and they can sometimes be completely pyritized as well. But note how, just saying, oh, it's replaced by pyrite is not enough. If you notice, sometimes it's replaced by these tiny microcrystals, quite cubic in nature. And sometimes you can start seeing formation of pyrite framboids. Now, there are different kinds of pyrites exhibit beautiful textures in the rocks. I've looked at about, 1,000 pyrite samples in the last, 6-7 years, and they come in all shapes and sizes. And there's a reason, there's a whole study on how pyrite textures can tell you the environment of deposition, etc., etc. So here's my very sophisticated chemical environment. I think this is far more complicated than anything Stephen or Nick or other chemists have showed you. So here's a chicken. I don't know why I chose the chicken. So we're going to pyritize this chicken here. I just chose the chicken because it's cute and I had to choose something. So we've got this chicken, and this poor chicken is dead, right? And so we form an environment of decay. This is really important because the decay environment will present its own microenvironmental conditions for the pyritization to occur. So this is really important. And we've got obviously sulfide, and we've got, so quite often these organisms are calcitic. And so the carbonate is slowly replaced by pyrite. And so there's a sort of dissolution of the carbonate shells, or whatever part of the, unfortunately, chickens are not made of carbonate. Can I just clarify that I do know that? This is just an example. But just think of a carbonate dissolution by an acidic environment, and then the decay products of the organic matter will also produce some hydrogen sulfide. And then immediately around the decay site, you need the availability of the ions. Now, important here is that this decay site has to be really contained, because if it becomes too euxinic, then the ion here will start to combine and form its own sulfide. It's only this diffusion of sulfide here from the decay matter needs to react with the ion to form basically the iron sulfide to begin with, monosulfides, and then eventually pyrite. So if the outside of the decay site, which is this area here, has a lot of H2S, then unfortunately this Fe will start to react and form pyrite. So this dissolution processes have to be very regulated and contained within the decay site. So here are two extremely complex chemical reactions that basically summarize what I've just told you. Okay, moving on. Fossil plants from Eocene London clay. So lots of experiments being done on pyritization of plant fossils in the lab to see how it all occurs, and what stages do they occur, what kind of textures are we forming, and you can see all kinds of examples. So for example, here you can see 3 different kinds of textures here. So this is the framboidal one. This is basically the mixture of framboidal and octahedral pyrite, and then you get to the more coarser one. So showing you what it looks like. So this is what framboidal pyrite looks like, and this is what you get: a mixture of framboids, get a mixture of octahedral pyrite, and then the really coarser one. So we've been, lots of experiments have been done on these on modern samples, and you can find this is a late Devonian equivalent of exactly the same thing. I mean, if you really squint, you might be able to see the framboids here. If you can't see them, squint harder. I'm sure you will be able to see them. And these are the cell, basically exactly what we were seeing in the younger samples. So when we find these things, it's pretty, we're familiar with what it looks like in the modern world. So it's easier for us to say, oh, that looks like a plant fossil, or we can do experiments and we can do this and that and all kinds of things. But when it's very tiny, because in Earth's history, as we go back in time, all your protocells and whatnot, they're very small. When that's being replaced by something else, we potentially have zero clue. So it's easy for this to be, oh, wow, spectacular. But when it's something very small, like say 10 micrometers, and this is a fossil. I mean, seriously, like if you came across that, would you really, I mean, it's only really, people who are experts in the field can notice things like that. So what if something like this ever got replaced by another mineral? And would we know that that was once a life form and now it's not? So that's the whole point of doing this research, is that doing it on known samples and then eventually applying it to the really tiny microfossils in the past. Because like I said, I've worked on, I don't know, hundreds of samples, and you come across all kinds of pyritic textures, and you quite often wonder if they're biological or not. For instance, this sample here, this is in Australia, in the MacArthur Basin, very close to a SEDEX zinc-lead deposit, where you get high levels of zinc and lead. And it's being mined for ages, but you come across some seriously weird-looking textures. Another example from the same area. So this is pyrite with sphalerite. And so you come across these things and you go, oh, they look similar to this and they look similar to that. Similarly, some more examples. And they have these various zonations. You can get all kinds of, like I said, shapes and sizes, and quite often they're overgrown with pyrite as well. As you can see, the 3 different layers of overgrowths here. And so we also, there are indirect ways of understanding what happened here. So we published a paper on sedimentary exhalated venting of fixed nitrogen. So we looked at nitrogen isotopes near the vent or close to the old deposit, and so like the trend was quite different as you move further away from a mineral deposit. And we said, oh, there was nitrogen available, and then we thought, oh, maybe that's why we start to see these biogenic texture. But that's it. It's a very indirect sort of relationship where we say, okay, there must have been nitrogen, so there must have been some life, hence these weird textures may be biogenic, question mark. If we don't have that question mark, reviewers get very annoyed because they want you to be ultra sure that they are biogenic. So I want to give you an example of why that's the case, because it looks biogenic. So, it could be biogenic, but we've got to, we're trying to find signs of life on Mars. We've got to have a better, lift our game a bit in the sense that we have to be sure whether it's life or not. I want to give you this example. Have a look at this picture, and then have a look at this, and then have a look at this. I mean, maybe you're all, I don't know, to me it looks pretty similar. And I'll tell you what they are. The first one is a micro photograph of heavy minerals in thin section of a tungsten. Don't worry what a tungsten is. It's not a biological thing. That's all you need to know. This one, where I'm not even going to try pronouncing the name, but it is biological. And this is an example of a chemical garden. They're very different. So my point is we need to have some form of a quantitative way of saying, well, it looks biological. Is there a way of being 100% sure that it is? And so that was the motivation, because I thought, well, I come across pyrite weed stuff all the time. Let's do something. Well, what else has been done? So there was a really nice paper by Marshall et al. I think it's 2017, and they imaged vanadium in microfossils. Because vanadium was part of this porphyrin molecule, and so you know, enhanced concentrations of vanadium could be signs of life. Fantastic, that's great. Let's do that. Let's see what other stuff has been done. And so yeah, you know, the microprobe allows you to look at element abundances. And so if something is biological, if there's presence of carbon, is quite often taken as a, oh, there's carbon there, so maybe that's biological as well. So there are various different ways. But element abundance may not be enough in my opinion. And I'll tell you why. So we thought, well, let's try on something we are 100% sure is biological. So here's an example of a non-pyritized ammonite, and here's an example of a pyritized ammonite. Now, remember the inside, the hollow part of the ammonite also has the fossil, but the ammonite shell will have also been replaced by pyrite. And I wanted to know if the hollow part of the chemical pyrite is any different to the outside of the ammonite pyrite. And I wanted to see, I mean, pyrite is going to be pyrite, which is FeS2. But what I wanted to see was the composition of the pyrite in terms of its trace element concentration. Was that able to give me some sort of idea? So obviously we wanted to look at the textures. We wanted to do all kinds of fancy things. Sometimes I don't even know why I'm doing it, but I do it anyway. Because somewhere, you know, conferences like this, you know, you come across someone and go, you could do this with that technique, and you're like, that's what I should have done with that data. And then, of course, you do what you're good at. So I have used the LAIC PMS for all sorts of things, and I thought I'd apply that to this program. And then statistics, another, my God, I do need AI for this because it's beyond my comprehension, that's for sure. Some of the statistical applications that we use on our geochemical data these days. And where's Juan? I hope you're here. I know you showed us the biological periodic table, and I was very, very upset to see just CH and OPS, because guess what? We need that also for life. Like I was feeling very left out that my trace elements were being excluded from your list. So you've got to be inclusive of the trace elements. So please do remember to include them. But no, the point is trace elements are very important for life. So we do need it. And so to understand how the variability, or more like the spatial variability, differs across the biogenic versus non-biogenic would be different. And with pyrite technique, we can do that. So that's what I tried to do. Basically summarizing my point, but I got carried away telling of one. But yeah, so basically trace elements super important. Even Superman needs it. Okay, let's go. Nano imaging traces. This is a paper I came across. People are using today in medical science to find the concentration of certain elements to outline the various parts of the cell of the brain. So it's not like I've come up with this genius of an idea. People are already doing it. Why can't we do that in our deep time samples is the question. So it's pretty cool that we can do these things. So my samples come from Southern England, where I've never been. My supervisor gave these samples to me, and we wanted to look at this. So this is a backscattered electron image of this particular ammonite here. And if you zoom at this part of the ammonite, it looks like this. So you can see these really fine microcrystals of pyrite. And if you further zoom in, this is also pyrite. So is this interstitial sort of mineral. They're all pyrite. But this pyrite looks, as you can tell me, unless I've gone mad, this looks very different to this bit here. And if you zoom in further, we've got, you know, similar sort of, basically a zoomed in version of this. And so similarly, you start to see differences in the texture. So that's the outer part, and that's the inner part. And then if you go, I've been shown the 5-minute science. I'm going to rush now. It's the same thing with the carbonates. If you use electron back scatter images, you can start seeing the differences in the texture. You can do, like I said, band contrast to see the difference in the crystal orientation. It's particularly good for carbonates, but then I wanted to see what the composition of the pyrites, are they different? So I did some spot analysis. So you basically analyze certain parts of the biological parts and the non-biological parts, and you see there's a difference in composition of cobalt and nickel, or selenium or zinc. You can choose any element that you like. I've just shown a handful of those. And then it's the same thing if you do it on the carbonate ammonites, you see some sort of difference in the geochemical composition. And then we thought of imaging it, because like I said, I didn't just necessarily want to do the abundance side of things. So we wanted to apply this technique called clustering on these geochemical images of these pyritized versus non-pyritized ammonites. So to show you what I've done is, this is step one, basically to get a compositional image. So I've imaged this area. When I say imaged, I've looked at this area for a range of trace elements that usually would go into pyrite. And I'm trying to see if the trace element absorption on pyrite replacing the biological organism is any different to the one that's got nothing to do with biology. So you can see there's a difference in terms of element abundance, but we wanted to further see if there's any sort of clustering. And we applied the clustering tool to this. And you see significant clustering in the biological area compared to the non-biological area. And you try that for a range of elements. And sometimes you see clustering, sometimes you don't. But ultimately, what you want is to see this line here, that's evidence of clustering, significantly above the 95% confidence interval. So that's essentially where I'm at right now. But what I would like to do next is to see if there's any difference in the speciation of the different elements that we're seeing. So clustering is one thing, but someone mentioned convolutional network, CNN something, neural network. I would love to try or apply something like that to my data as well, just to figure out what's going on. This was just an because it looked cool. And yeah, this internal disk has this clustering. Does that mean I found the solution? No, I don't know. This is just like I said, I'm new in this game. So I'm more of a scum than you are. And I'm just trying to figure out what's going on. So we've got some beam time at Soleil in France. So we would be looking at some of the speciation on these pyritized fossils. But yeah, there's a very cool paper that's recently come out where they've basically come up with a range of sort of textures that look very biological, but they can just be synthesized in the lab with no biology around it at all. So they've they have a different sort of decision tool system, again, a statistical term for assessing biogenicity that takes into account the geological history. And they've given 2 options, but usually it's more than 2, obviously. So it's very case specific. You know, it's not like one rule applies all. So what applies to pyrite may not apply to apatite at all. And so that's something we need to take into consideration. That is not me climbing over the papers. Just very quickly before I end, and I'll literally take half a second, is that a lot of this orthogenic mineralization depends on the fact that these decay occurs, and the decay is regulated by microbial organisms. This is how much we know about our microbial organisms. 2 out of 3 domains of our life is microbial. And we really need to know a lot more before we can understand a lot. So I'm not shy of admitting how little we know, and I'll end on this slide here so we can find more about this. Thank you.
We could all listen to you forever, to be honest, but one quick question for you, Johnny.
Thank you for bringing mineralogy and passing me in one of the slides. I have a question on magnetic active bacteria. So kind of information seeking question. So what's their role, a significant role in biochemical element cycling, and also are they widespread across the domain bacteria?
Are they widespread across Roman bacteria?
Domain bacteria.
Domain bacteria.
That shows the
Yeah, so I have no idea about the second question at all. So I'm not a biologist, but I will tell you that the role of the magnetotactic bacteria is, I think, mainly to do with chemotaxis. I think it's usually used for alignment or positioning because it aligns itself in a certain way and it's attracted to nutrients. So it helps the bacteria with its positioning.
Maybe one question as the next speaker comes up. All right, we'll have two good ones, Josh.
I was just wondering, it's very interesting, it's sort of like you felt growth of the pyrite. And in the earlier in your slide, you had a bit where it's like there's limitations in terms of the resolution of the stuff, the size of the pyrite crystal. I was wondering, like in this field, do people think that like is the size growth of the pyrite crystal, is that limited by the sort of like a boundary or sort of like a cellular organism, the size of it, or is it sort of is everything sort of being seeded at once and it's more of like a kinetic limitation of the size of the pyrite crystals?
I think it's mainly to do with the detail of the information preserved. So I think because apatite has a much lower size of the crystal, it's able to preserve more detail. Whereas in case of pyrite, a lot of the detail is lost and much more of the coarser details of the fossils are preserved. But again, the problem is quite often the H2S can get really high because it's an anoxic environment and if around the decay site, so then you'll get no detail essentially, and you'll have a massive pyrite precipitation around. So that sometimes you can, it's quite possible that you preserve no detail whatsoever if the chemical conditions change too rapidly. So Simon?
Yeah, thanks. Fascinating talk. Just a quickie about the Ashtragods and the controls on whether it's cubic or rhomboidal pyrite.
So why it's cubic or why it's, it's knowing how rhomboidal and cubic crystals of pyrite form in varying sedimentary. I think it's an EHPH. I can't think of, like, in a chemical environment, you can have both together. Why would it necessarily? I mean, it could be possible that certain organisms might have some sort of organic template that's sort of inducing rhomboidal pyrite formation. But that it would probably suggest it's forming more sort of greigite kind of iron monosulfides as iron sulfide precursors instead of sort of non-greigite iron monosulfide. So that would probably induce more rhomboidal pyrite precipitation, and that would the others would. But the thing is, they can both occur with biology or without biology. So there's no. It's just a chemical parameter solely, I think.
Cool. So let's thank you and joining one more time. So our last three talks are by, I think, the most junior presenters in this whole conference. We have a master's student and then an honors student. So let's welcome Emma Nasezova from University of Auckland. Thanks.
Hello, everybody. Thank you, everybody, for sticking around, even though it's the second to last talk on the last day of the conference. Love that. Actually, I'm really excited to be following Indrani today because she has set me up so unbelievably well to start talking about my biosignatures, and she's explained all of the horrendous biology really nicely. So hopefully I won't confuse you even more because your brains have already been seeded with that great information. So today I'm really excited to share with you some really awesome research that's coming out of Auckland University, in Mike Rao and Kathy Campbell's research group, studying digitate sinters, which we've been doing for a number of years now. And as we all know, oh, hello. Okay. Oh, okay, we're ready. Cool, exciting. So I'd like to take everybody on a little journey today. Welcome to Mars. These are the famous Columbia Hills that we've been hearing so much about Martin did a great introduction of them the other day, so we won't go too much into it, but essentially in 2007, the NASA's Spirit rover found deposits of opaline silica, this kind of nodular structures around home plate, which is this volcaniclastic feature, also in association with sulfur-rich soils. And generally this, on Earth, these kind of environments are interpreted to be from hydrothermal activity. So this is telling us that we're finding deposits of silica on Mars in a hydrothermal environment, which is super exciting. And why is it so exciting? That's because, as you can see, these kind of really finger-like structures are found in some of the imagery from the deposits. And on Earth, we find these finger-like structures in hot springs, and they are just teeming with life. So they, some of them are filled with cyanobacteria, some of them have other different kinds of microbes in them, but essentially the general structure of a digitate stromatolite is laminated silica and microbes forming these finger-like vertical protrusions in hot spring environments, which is super exciting and is, it basically leads us to ask the question, is there microbes in the Martian silica, since it's both morphologically and mineralogically very, very similar to the stuff we find on Earth? As Martin introduced us, this is the target for the LifeSprings Mars Developing Sample Return Mission, which is very, very cool. So, right, I have been studying these for a very long time, not that long, but long enough. And essentially, this whole research that I'm doing now came about from my honors research, where I was investigating the density differences and the kind of mechanical properties of these structures. Shameless plug for the paper that's in review currently. And essentially, we found that the porosity that we can see in these laminae is controlled by the microbes that form them. So the cyanobacteria and the diatoms and stuff that form those stromatolitic laminae is what controls the porosity. However, as we can see, the density is, there's quite a difference of variation in density. And I kind of asked myself, you know, how much of that is actually chemical variability playing a role in there? So that's kind of what led me to start investigating my master's. So where does digitate sinter form on Earth? Well, in a variety of different environments, but always at the air-water interface. So this is a subaerial deposit forming at the boundary of the hydrothermal fluids and air. Around the rims and in the discharge channels of hot springs. Essentially, we always need to have some sort of repeated cycle of wetting and drying, evaporation, and things like that to deposit layers of silica on top of microbes that are colonizing the surface and actually templating that deposition of silica. So those layers build up and up and up, and then we end up with these vertical protrusions, these digitate structures, which are outlined here, and they form in all hot springs of all different kinds of pHs, all different temperatures, doesn't really matter, they all form them. It's great, we love it. So, thank you, Andrani, for setting me up excellently for this. How do we detect life in sinters? So essentially, there's three main types of biosignatures, I guess, that we characterize in the literature in sinters. Morphological biosignatures, which is things like cells and cellular products. So this is an image of a microbial filament from alkali chloride sinter. So it's anything that is a cell or is produced by a cell. You can also have organic molecules, which here I've got an image of Raman identified kerogen in Jurassic geyserite, which is a type of sinter for those of you unacquainted. And you can also have metabolic biosignatures. So this is what Andrani talked about, but I'm going to focus more on the microbially induced permineralization ones. So essentially indirect or direct microbial precipitation and sequestration of metals, which still technically falls under the metabolic category. So my thesis focuses on the organic molecules and the metabolic ones because the morphological ones are pretty well characterized in literature, but today's talk is just going to focus on the metabolic biosignatures because I could be here for a long time and we don't want that because we want beers. So, right. First of all, before we can do any sort of biosignature characterization, we have to characterize what is biology in sinters. And essentially, this is mostly a function of pH. So temperature is often thought to be one of the most controlling parameters, but a lot of research that's been coming out of the Topol Volcanic Zone, the 1000 Things Project, has actually figured out it's more pH that controls it. So in near neutral digitate centers that, whether it's low to mid temperature, it tends to be filamentous cyanobacteria. In this case, we've got some Calothrix filaments here. Whereas in the acidic digitate ones, we tend to find some diatoms, which are a type of algae. These ones specifically are pinnate diatoms, so they look like little grains of rice. And we also find cocoidal algae and various different bacteria that I have not identified. But essentially, these are bacteria and algae that thrive in acidic environments. Cool. So we know that cinders are full of life. That's exciting because we've been banging on about it, but you've been taking my word for it. So we've shown it now. Now, looking at the trace element distribution and the major element distribution, essentially, I did two techniques. I did WDS, and then we were lucky enough to get some beam time at the Australian synchrotron earlier this year, and to do some X-ray fluorescence mapping. So that went along hand in hand really quite nicely because the fluorescence mapping was quantitative and the WDS is just qualitative distribution. But essentially, as I was, making my maps, scanning along, I kind of noticed all of a sudden that these areas of microbial enrichment that I could see in the petrography and in the backscatter images, they were the areas that were sequestering elements like aluminium and iron. And that was really interesting to me. So I managed to prioritize those areas for X-ray fluorescence mapping. And we found also, again, in both of the samples that I had that were from alkali chloride hot springs, all of the areas that had the microbial filaments visible were just pinging in metals. We had gallium, we had calcium, we had iron, we had arsenic, we had strontium, we had rubidium, we had everything. So that was really, really cool to see that. And yeah, it was it's already been observed a couple of times in literature, but to observe it in samples in the Topol Volcanic Zone that are this young is really exciting. So zooming in a little bit closer, it's not just a homogeneous distribution of metals, in vaguely microbial areas. You can see that the metals are actually just discretely outlining these filaments here, which is really beautiful to see in all of the samples that I found. And essentially, another interesting thing that X-ray fluorescence tells us is that there's several different layers of filaments that are living in different growth orientations here. So a little bit of a background for those of you who are not familiar with fluorescence. This technique that we're using, the lighter elements fluoresce from the fluorescent rays can only travel a very short distance. So you're only capturing the top few microns of sample, whereas something like as heavy as strontium, those rays can actually go very far, and so you can capture them from up to 700 microns within the sample. So these samples are only around 200 microns thick, so that's, you're not getting 700 microns worth of strontium there, but you're getting about 200 of it. So essentially what it's showing is that in the calcium, which is the 15 micron one, the very surface level, and the strontium, which is the deepest one, you're getting transversely oriented filaments. So filaments that are kind of sticking out at the screen at you, and they look like these little donuts shaped things here. Whereas in the gallium and the iron and the arsenic, those kind of around between 70 to 300 microns, they're lying down, they're like longitudinal. So essentially what I think is happening here is that you've got some filaments that are kind of oriented perpendicular to those, sticking straight out at you. The calcium is recording the front intersection of those filaments with the surface, and then the strontium is recording at the back. And then the filaments that are longitudinal and lying down, those are the ones being recorded by the gallium and the iron. I just thought that was really cool. So I'm not a biologist. I can't tell you what that means in terms of their environment. I've heard that there's some sort of correlation between the time of day or night and the orientation that they're in. So that could be something I'm investigating, but I'm just a humble geologist. So yeah, again, humble geologist explaining bacteria to people. I'm sorry to anyone who is a biologist in this room. But essentially, this is my little graphic of what cyanobacteria look like. So essentially, each one of those filaments, that's not one organism, that's like 50, all little individual cells joined together in a trichome that are surrounded by a sheath made of EPS, which is basically polysaccharide, sugars, proteins, and lipids, all kind of jumbled up in this little goopy situation. And it's excreted by the microbes, they make it, and it has a bunch of protective purposes. But that's the general kind of structure of what the filaments look like. So we can see that in the samples in the petrography. And also when we look up closer at the, at the X-ray maps, we notice that the elements are not distributed also throughout the entire microbe. They're concentrated around the sheaths. So that mucilaginous, mucousy, goopy stuff that we talked about earlier, that's the happening place. That's where all the microbes, the metals are being sequestered. And so I was sitting there wondering to myself, why is that? You know, that's kind of, it's a bit weird. So I was trawling through literature, as you do when you're a master's student. And essentially there's been a bunch of lab work that's been done that prove that these sheaths are actually super reactive, but to specific metal cations. So they have negatively charged surfaces on them that actually sequester metals, which there's a bunch of reasons why it could be. It's kind of, there's not really an established answer as for why, but essentially it's thought to be some sort of preservation situation again, which does question the idea that it's just, you know, a passive process, because if there is a benefit to it, couldn't you make the argument that it's controlled in some way? But, you know, something to ponder. And Johnny's nodding. That's exciting. So yeah, essentially beautiful sequestration of metals around the sheets of these alkali chloride cyanobacterial filaments. And I even have a reason for it in literature. So awesome. So the other good thing about this is that the sheaths, because they're so readily mineralized, are the thing that's preserved the most in the geologic record, which also is a good news for us because on Mars, those are the things that are most likely to be produced. So if there is this kind of biological fractionation of metals happening, it's the most likely thing to be preserved. So number crunching for the geochemists in the room that want actual concentration values. Here we go. Essentially, we are finding enrichment in these trace elements at orders of magnitude higher than what it is in the fluid. So for example, for Altatio, the calcium is at about 30 ppm in the fluids, and it's at 1.36 weight percent in the sample. So this is an incredible like fractionation that's happening here. And I'm not really in a position to kind of comment on how much of that is biological and how much of that is just a, you know, chemical mechanism. Maybe the silica is, you know, there's just abiotically kind of binding some of this calcium. I'm not sure how much of that enrichment is biological or chemical, but you can clearly see that there is a strong biological component to it, which is really exciting. So yes, what about the acidic ones? So we've spent a long time talking about alkali chloride centers that are full of filaments. So as we saw earlier, the acidic digitate centers are mostly composed of diatoms. So essentially you get the same sort of stromatolitic laminae, but this time instead of silica filament silica, you end up with silica diatom silicon. And the diatoms, I didn't find any sort of convincing evidence that there's any sequestration in them. To me, that looks, you know, pretty blah. But yeah, so diatoms, they are very different organisms to bacteria. Obviously, they're algae, so they have, and these ones specifically are really cool because their cell walls are made of silica. So they actually absorb silica from the fluids that they're living in. These are subaqueous organisms, and they precipitate silica to make their cell wall. And because we're not seeing any evidence for biological sequestration of trace elements in the silica, I'm thinking it's just because they're absorbing trace elements at the same rate that the silica is being deposited from the fluids. So the application of these exciting biosignatures is probably only limited to the cyanobacteria that we're seeing, so alkali chloride centers. So yeah, in conclusion, I think I found some exciting metabolic biosignatures based on this definition of a sign of microbial metabolism or active passive sequestration. We can argue for years whether it's active or passive, but regardless, it's there, you know, you can see it's being sequestered. So for my purposes, that's good enough. Yep, here they are. We've seen aluminium, calcium, iron, gallium, rubidium, and strontium in cyanobacterial sheaths in Altatio, as well as the other alkali chloride center that I studied. Specific to alkali chloride centers, so nothing found in the diatoms yet. And the application of these biosignatures into the deep time, both on Earth and then also on Mars, would depend on their preservation potential. So more studies need to be done going forward about how this actually translates into the deep time on Earth to see how it could potentially apply to Mars. However, we do know that the silica on Mars is not actually crystallized at all. It's still in opaline form, opaline silica, so it hasn't actually undergone diagenesis. And hopefully any primary features that were in there will still be there. So exciting times.
Any questions?
Yep.
And great talk. I was just wondering in terms of you were mentioning that it's sort of the bottom system based on the brutal sheet.
Mhm.
And then there's like everything that's part of all. So even the mineral that you're looking at is most specific findings. Like mineral. So I think my question is there sort of like say are these metals or complex to say silica in a rock, or is there evidence to say carbon cations, like if you look at carbon concentration, does that all get wiped away by silica over the biomineralization process, or is there like it's like I would think that like in the biological system, what would it say like okay, the sheet would be maybe like the heads of fatty acids or like lipids or something like that, or something like chelating group.
I think they're like carboxyl groups or something. I didn't, yeah.
But in this system, you've got silica. And I was wondering if there was any like this, or if it's just not achieved measure. Is it like do sort of account the anions or chelating anions that would have been in the biological system? Do they get wiped away in the biomineralization process?
Not really, no. So in some of the Raman work that I've been doing, I've actually been able to characterize some of the organic compounds that are still in there. So obviously through deep time, yes, they are wiped, but in the stuff that I'm looking at, this is modern, it's been plucked out of the hot spring a couple of years ago. So it's actively forming and those compounds are still really well preserved. I've not found any evidence to show that the organic compounds that I've found are chelating metals specifically. There's nothing really to say that. But it definitely does happen in these kind of systems. And I think, yeah, there's some sort of property about the lipids and polysaccharide complex thing that has these sticky carboxyl groups that are sticking out and chelating the ions. But I'm not sure which exact compound it would be. That's not really something that we've been able to detect preserved in these samples. Okay.
So when you have enrichments and parts per million, presumably that means you also have elements that are depleted.
I've not, I mean, depleted relative to what?
Well, I mean, whenever you gain something, you have a million parts. And so now they've got more parts per million of gallium, so you must have less parts per million of something else. And the question is, so if you're trying to come up with a mechanism as to why you concentrate those, it's useful, it might be useful to have a statement about what elements are being depleted. That way you have a mechanism because you have what's. So what elements are less abundant there in the background?
Yeah, it's a good question. The thing with the X-ray fluorescence is that it only measures what's there and what's not. So if something is depleted to the point where it's not there at all, like I can't, like the technique itself won't give me a number for something if it's not there.
Well, the number would be 0.
Yes, yeah, no, that was a stupid answer. Yeah, it's something to think about, I guess. Yeah, I hadn't thought about phrasing like that, so I might go ahead and do that.
We got one more question. All the next speakers.
Just follow on from that, really. We can do the geochemical analysis of fluids, of the hot spring fluids, and see the more concentrations of various elements. And so in some cases, yes, there are elements we know are in the hot spring fluids that aren't dissipated out in these things. So there's definitely some selection of trace metals that makes ratios that are in these deposits which you wouldn't find in just an abiotic.
Sure. But I guess the question of what parts per million means, per million what?
Well, volume of the analysis.
It's not atoms per total number of atoms, it's grams per total numbers of grams. No, not grams of gallium per million grams of second. Yeah. I mean, the fluorescence is telling you something that you actually got how many molecules are there, I suppose, if you have some standards, but so you don't actually know, you know that there's a lot of gallium there, but you don't know how much gallium is ratio to per million of something else.
Okay, I guess. Yeah, you can continue that discussion on the plane ride. But yeah, let's thank everyone last time. And for our last speaker, Kushi Daga, she's an honor student. So yeah, there's.
Hi, I'm Kushi. I'm part of Anna Wang's lab, and I don't know if I should be grateful or confused and upset that I'm following those series of wonderful talks. So bear with me, I'm going to do this a little differently. All right, so for a second, let's leave this lecture hall behind. Come with me on a journey through time. if we get the slides to move. All right. 300,000 years ago, we had the first of our species, the first Homo sapiens. And if we go further back, 252 million years ago, the first dinosaurs, right? But we're not done yet. One step further, and we had the first fish walk onto land, and yet, this is still not the environment I want you to imagine. The Archean Eon, all right? Part of the Precambrian period, 4.6 billion to 546 million years ago. It's one of beautiful toxicity. We're imagining a period, an environment that has highly detrimental rays of ultraviolet radiation. Surface temperatures can fluctuate anywhere between 0 to 40 degrees Celsius. In comparison to today, the average temperature, surface temperature, is 14 to 16. That's a big difference. During this time, there was a period of cratering known as the late heavy bombardment, and this meant that meteorites and natural geochemical processes were creating an abundance of prebiotic molecules, all right? And the mantle was allowing for terrestrial and shallow marine siliciclastic accumulation. The atmosphere had no free oxygen, meaning the first forms of life needed to be anaerobic, needed to be protected from these high UV rays, and it needed a rich nutrient supply to thrive. Here, that's why we find that the belief is life probably originated in hot spring pools or near hydrothermal vents. And the geological setting I'm looking at are hydrothermal pools. Okay. Thank you so much, Indrani and Emma, for setting this up so beautifully for me. So I'll fly through the next few sections. So geothermal pools, what are they? Well, these are these natural accumulations of aqueous solutions, and they're heated naturally by the earth. They can range anywhere between 17.5 to much greater than 90 degrees Celsius, and their pHs range between 1 to 12 due to natural alkali and acid chlorides. So this means that there is an abundant supply of molecules and chemical synthesis, and it induces a natural microclimate, which allows for cycles of evaporation and precipitation. This natural microclimate is what allows for silica deposition. However, these environments are inseparable from chemical traces of life, making it difficult to tease apart non-biological components from biologically associated components. I think we can all recall Indrani's beautiful little balls that have the spikes. She really told us how important it was to pick these ones apart. So we've all been looking at the silica nodules that kind of look like digitates on the surface of Mars. Now, silica accretion is a key marker of complex aqueous chemistry, but the nodules resemble sinter that are found on Earth in areas that are only found with the presence of life. So the ambiguity surrounding silica deposition allows for interpretation bias upon these Martian masses. And this is because we don't quite understand how important biogenic matter is in this process. So we're questioning whether there was the ability for silica to create 3D structures prior to the rise of biota. And this is a beautiful quote. I don't want you to read the whole thing, that's all right, but it's a beautiful quote that summarizes how important interpretation bias can be across so many different fields. So this is from an archaeology paper, and it really details that when we're reconstructing like archaeological landscapes, we tend to apply features that we draw from our own environment. And this can create a preconceived notion of what structures may be, represent, or hold value to us. And so this is what we want to minimize moving forward in astrobiology. There is some key previous work, especially Dr. Kathy Campbell and of course Emma, and they've really looked at the environmental conditions that affect silica deposition. So we know that the air-water interface is greatly responsible for the way molecules aggregate. And we know that it can differ due to conditions like, actually, I'll go into that a bit later, but silica accretion on these slides have been placed into hot pools. So glass slides were placed into hot pools, and they were left there for a few weeks for silica to accumulate. They were then studied, and it was determined that sinter physiology can change due to things like splashing, wicking, and other physicochemical aspects. Now, this is really important to note that on the right, we see laminated sinter, and it developed in a pool with changing water conditions, but a fairly stable temperature. And that's compared to spiculose sinter, which was generated in a stable water level region, but has a wide range of extremities in temperature. Okay, so what do we understand so far? We know that sinter has only ever been identified in areas of flushing microbial activity. This can be seen in archaic stromatolites and extant hot pools today. We're now aware that there is an ambiguity to determine how important the biogenic matter is for this formation to build up as a macro structure. So my research is going to be looking at, well, can we assess what abiotic chemical factors can affect these? Okay, so I focus on wet-dry cycles as a mode of silica deposition. The cycle starts with the wet phase, when water levels are replenished by rain or deep groundwater. And this oscillates as temperatures rise or fall in the natural microclimatic vicinity. And as the evaporation rate increases, there's a low water content. This means that the air-water interface lowers towards the bedrock, and it deposits any silica and other bound material that have been trapped. This is what entombs biogenic material. So since wet-dry cycles are concerned with the suspension of dispersion of molecules through medium, they are subject to a series of microfluidic charges and forces that affect the molecular behavior. So with a major in physics, I'm looking at the very first steps of silica deposition through a phenomenon called the coffee ring effect. Anyone who has spilled coffee before is familiar with this effect. It genuinely means that we have an understanding that a droplet experiences capillary flow. So when you drop your drop of coffee, what you might see is that particles are driven towards the edge. And this causes particles to become pinned on the outside of the droplet as the liquid dries, as the liquid works. As the water evaporates, this edgeward flow causes more material to be deposited on the outside rather than as a film evenly across the surface. This theory is highly applicable to suspension of silica molecules because it could help us understand why particles are depositing where they do. So the physicochemistry of colloidal deposition is what I'm particularly looking into. So particles that are suspended in droplets can be manipulated to accumulate or disperse based on these surface conditions. Factors can be altered by things like pH, temperature gradient, addition of fatty acids or polymers, to name a few. So in my experiment, I'm exploring deposition morphology as a function of some of these factors. And I seek to observe the prevalence of the coffee ring effect in deposition because it can really tell us about the charges between these silica molecules. So the method is I'll be depositing different silicate solutions onto a hot plate through a syringe, and both are at a constant rate for repeated wet-dry cycles. All right, so some interesting results that I have so far. At the low pH, I want you guys to look for this spotty cloudy, this signature cloudy pattern across my samples. Can you see them? If you say that you can see them crisscrossing all across the slides, overlapping, you'd be right. So at a low pH, the coffee ring is less apparent. This means that there is a more even film of deposition. We see a signature pattern of cloudy or undulating textures, And we see delta or branching type patterns as well. We also can observe microcrystalline structures under microscopes, which are suspected to be sodium chloride due to pH adjustment. We already observe the formation of structures with complex morphologies that could possibly indicate the development of knobby or rounded textures. The mid-pH zone. We notice that the coffee ring effect becomes a little bit more noticeable. We have significant vertical growth and the edges begin to build more mass. We remember that at a low pH, we saw those signature cloudy patterns, right? Well, you don't see them here. The droplets are more spread across the slide in the low pH rather than here collecting in a more centralized zone. So there is also a mottled or granular pattern we can see that's sort of emerging. And there's a high crystal content as well, cubic, almost similar to halite. It is key to notice that a simple change in pH can drastically affect the deposition morphology. And those are just those crystals highlighted. And those are on an upright microscope and they're not too zoomed in, so they're actually visible to the naked eye. Finally, at a high pH, we notice that there is major evidence for the coffee ring effect with drastic upward growth growing at the rims. A strong granular pattern develops, and there is slight evidence of the mid-pH. And there are sort of these linear stress fractures visible on the middle sample, and these are visible to the naked eye. Under the microscope, there is, again, crystal formation visible. However, because these are pure silica solutions, they're untouched by NaCl or any pH adjustment, we actually see a very different kind of crystalline structure, almost appearing more jagged or like a sharp fractal. And you can see that there. Okay, let's do a quick comparison before we go to stage 2. So the low pH is characterized by the undulating edges, whereas the mid pH and high pH, these textures disappear. The coffee ring effect becomes more pronounced as we increase pH, telling us that the capillary flow is more noticeable at these high pHs. And these samples with high pHs have more vertical lifting, but we want to also question how important is the coffee ring effect in developing sinter? Because if we have hollow structures, are they really stable enough to build silica nodules or silica digitae? The next step is determining the effects of organic molecules. So the same samples as earlier, except this time I'm varying the solutions with things like organic polymers. Glycerol and citric acid are actually known to form like a sticky branched polymer from a study in 2019. And decanoic acid is a fatty acid, which is known to decrease the surface tension of water. Hint, hint, that's soap. All right, so here are some primitive results for this stage. So staying within the high pH range, we have pure silicate samples on the right, and decanoic acid and citric acid and glycerol on the left. You can see that these organic molecules are seen to alter the pattern of silica accumulation. However, there are very stark similarities between the two. You can see things like granular accumulation and thick rim deposition. So decanoic acid at a high pH exists in a soap-like phase. This means it's lowering the surface tension, meaning like the molecules are weakly attracted to each other. So perhaps we could look into decreasing surface tension or minimizing the coffee ring effect for the future. And those are just those same two samples, but taken from the side, and you can see how much vertical lifting there is. Yes, so in hot springs, we know that there is ample energy supply, whether it's like thermal energy converted from kinetic energy, things like bubbling and splashing. So these are all factors that also could be affecting the way that these molecules aggregate or accumulate. Some features that I'm keen to look into in the future are things like cracking or porosity. I want to conduct several repeat experiments. So I can take a look at things like stress fractures that radiate outwards. That kind of insinuates a cracking regime as a part of silica drying. And there's a few previous studies on this phenomenon. So we can also see there's like this unique hatch pattern that sort of shows through, and they kind of look like microscopic stress fractures. So these are things that I'm really keen to look into. Okay. And some more interesting observations, just because I think these pictures are super, super cool. The first image is kind of a positive growth within a bubble. So you can see inward pointing fractals down in the corner. And the second image is kind of a similar thing, but on a larger scale. There are more pronounced salt crystals in that one. And there's like this dagger incrustation. That image is actually outward radial like linear fractures. So for future characterization, I hope to look into like quantifying the vertical growth and the profile of these samples from various directions. And elemental analysis could be really, really beneficial to looking into whether it's silica or other material. Also looking at like opaline silica versus amorphous precipitation. And I want to also examine primary silica size, particle size using SEM, and like compare them to other structures that we see in real life hot springs. So what can this mean on a larger scale? Is the droplet really a perfect representation of a hot pool? Well, the experiment so far does evidence that A, microbes aren't the only thing that change how silica deposits, and B, it also drives that there's hard evidence that the early stages of silica deposition already change morphology due to solution conditions. So while it's not a perfect microcosm, it does allow us to dive deep into some of the many processes that are occurring in this hot pool. And we notice that physics and geochemistry is actually a very important part of this process. So in the future, we look forward to more experiments that bring droplets together on a large scale. People like Megan have already begun on this, and she will be looking at things like splashing and orientational aspects. So you might have seen her poster on Wednesday. It's fantastic. So building on these theoretical blocks is going to be really beneficial to like untangling biotic from abiotic structures, and perhaps it can help us characterize the difference between the two. So is sinter really a biosignature? I don't really know. There's a plethora of combinations that we could be trying, minerals, chemicals. It's a very preliminary experiment, but it does prove beyond doubt that this is something we need to be looking into, because simple morphology is not enough to claim a sign of bias. Thank you so much. I just want to thank Anna and the Wang Lab for all of their support. Thank you so much to the ACA for this incredible opportunity, and to everyone in the audience that's here to listen to me today. Take your questions.
You said that hot springs changed a lot in temperature. Would you look at temperature studies, and is the effect that you're looking at the coherent, is that like affected by temperature?
Yeah, temperature gradient can be very important to these kind of situations. Just in the space of an honors year, I actually can't vary so many factors at once. So I'm already changing like mineral, like organic molecule composition. I'm changing pHs. So also I already have like a variety of solutions that I'm trying. And while it would be wonderful to look into that, I think people like Megan will be taking that forward. Yeah.
You mentioned that it's difficult to differentiate the chemistry of hot springs, biological and abiotic. Are there any ways you can attempt to mitigate that with like when you look for freshly active springs or salmon from particular locations?
Yeah, that's a really good question. So I think, so the problem is that because these hot springs are naturally occurring in the environment, they're already receiving so much like there's already bacterial action in the vicinity. So they're already contaminated with biotic or like organic molecules that sort of like it takes away that abiotic aspect. And if we were to go to hot pools, we can't really like bleach it. You know, a lot of these hot pools are in protected locations, and to wipe out thriving life within a hot pool would be ethically questionable. So while that would be fantastic to do and like restart life in a hot pool. I just don't think it's something that's like possible at the moment. Round of applause. So thanks so much to all the speakers that have just given us like a fantastic last session and last day. I'm going to just give some brief closing remarks before letting you all go and party somewhere. So, right. So Albert started this. I'm here to round it out. So for those of you who don't know me, I'm Anna. I'm a physicist pretending to be a chemist, but I love astrobiology. So firstly, I have to thank everyone who made this possible. For lots of good feedback about how good the conference was, and we all had a good time, but these were the people getting stressed out, making sure we all had a good time. So, you know, Albert had lots of sleepless nights, and also the rest of the organizing committee went into all the fine details, like from catering to making sure posters and everything were running smoothly. So let's give them a huge round of applause. I'd say that if it was just me and Albert, we would have dropped a lot of balls and there would have been no one to pick them up, but we're very lucky to have this whole team. The next part is thank you to all of you. Some of you have traveled from very far away and arrived last minute, have to leave really quickly. And, you know, I always think of astrobiology as like this funny place where it's like we're all going to use a music analogy. We're all like playing instruments, right, in our homes with our own little like sections. But these astrobiology conferences are where we all come together and you can start to hear the music happen, right? There are lots of big questions. We're all dreamers in some sense, but it's great to bring this community together regularly. And so thank you all very much for participating and coming here. Next, just if you're still in Sydney for the next few days and not going to Western Australia, there are lots of cool free things happening. And so the Powerhouse Museum, which is our Design, Science and Technology Museum, and Museum of Industry is open. It's close to the city. And it's free, Thursdays until 9. And tomorrow, the Museum of Contemporary Art has this wonderful terrace. We have a view of the Opera House, we have a view of the Harbour Bridge. There's usually a DJ and cocktails and nibbles. And so that's a cool thing to check out as well. And there's also lots of Indigenous artwork at the Museum of Contemporary Art. You're also here during Vivid, which is like our biggest winter thing. And so check out the lights. So you can start in Darling Harbour, walk all the way to Circular Quay in Barangaroo, and see lots of iconic structures being lit up by artists from around the world and also from Australia. And the last thing I will say before rounding out this conference is that our hero Joe has agreed to host the next AAM in Hong Kong. And so this is fantastic news. So mark your calendars, mark your diaries. He's picked around November because of, you know, holidays and also good temperature in Hong Kong. But, you know, this is a really important community to bring together across Australasia, and it'd be great to have it in Asia. You know, we look forward to seeing all of you there. So thanks everyone and have a great afternoon. Some people have a plane to catch. Right. For those of you who are staying on campus, the lounge is like a rooftop bar thing that faculty can go to. If people are interested, we can lead a group of people there. Otherwise, there's also the Roundhouse Pub.